welcome you to the topic of Masterclass on obesity and cardiometabolic diseases. This event was organized by Metabolic Disorder, Cardiovascular, and Aging Research Cluster (MPA) as part of the 6th Annual International Conference and Exhibition on Indonesian Medical Education and Research Institute (ICE on IMERI), which held from 5 to 14 November 2021. With the gathering of academia, researchers, business entrepreneurs, practitioners, and policymakers in this event, there will be updates on the latest knowledge related to obesity and cardiometabolic diseases, including pathological mechanisms, prevention and therapy, as well as improving the quality and quantity of appropriate research that produce breakthrough ideas that can be applied in community service through collaboration within and between institutions. Excellencies, participants, ladies and gentlemen, we are now going to listen to the opening speech delivered by the head cluster of Metabolic Disorder, Cardiovascular, and Aging Indonesian Medical Education and Research Institute, Faculty of Medicine, Universitas Indonesia. Please welcome Dr. Diki Levenes Tahapari, Specialist of Internal Medicine, Consultant of Endocrinology, Metabolic, and Diabetes, PhD.
Thank you, Mbak Tika. Good morning to all distinguished speakers, especially Professor Shimizu from Juntendo University. Welcome to IMERI, to Indonesia. Also, Professor Yoshida from Juntendo University as well. I also would like to welcome Professor Sidartawan Sugondo, also Dr. Bambang Widiantoro, Dr. Farid Kurniawan, and Dr. Agian Jevindo. So in this session this morning until afternoon, we will hear presentations sharing experience of research and also clinical experience in the field of obesity and cardiometabolic diseases. As we all may know that Indonesia is facing increasing numbers of cardiovascular diseases. And one of the main drivers of the increasing numbers of CVDs is actually increasing of obesity. Based on the latest basic health research, Indonesia had around 1/3 of adult populations were obese. So this may lead to increasing risk for cardiometabolic diseases. So in this masterclass, we hope we can learn a lot from all the experts today about the basic science of obesity and cardiometabolic diseases. But not only the basic science, we will also learn about the clinical implications of the mechanism, and then we hope we can then reapply those in our clinical practice. So this masterclass encompasses a really wide spectrum from basic science into clinical applications, as well as from the epidemiology perspectives. So we do hope all the attendees could learn a lot from the speakers as well. And the other way around, we hope that the presentation from all the speakers can incite a fruitful discussions between the attendees as well as the speakers. So without further ado, I would like to again thank all the speakers and all the chairpersons of this masterclass. And I hope you will all have a fruitful discussion and hopefully also open a new collaborations ahead. Okay, with this, I open the sessions. Hopefully you all have a fruitful discussion. Thank you for your kind attention.
Thank you, Dr. Diki Levenes Tahapari, for the opening speech. We are now continuing our meeting to the next agenda. The first session, current trends of translational research in metabolic diseases from basic to clinical practice. The first session will be moderated by Dr. Diki Levenes Tahapari, Specialist of Internal Medicine, Consultant of Endocrinology, Metabolic, and Diabetes, PhD. He completed his medical studies program from MD to subspecialist at the Faculty of Medicine, Universitas of Indonesia, and earned his PhD at Leiden University Medical Center. Now, he is a staff and lecturer at Division of Endocrinology, Metabolism and Diabetes, Department of Internal Medicine, Faculty of Medicine, University of Indonesia, Dr. Cipto Mangunkusumo, National Referral Hospital, and also as Head Cluster of Metabolic Disorder, Cardiovascular, and Aging, IMERI FKUI. Please welcome Dr. Diki Levenesta Pari.
Thank you, Mbak Tika, for the very kind introductions. So we will begin our first sessions this morning, entitled Current Trends of Translational Research in Metabolic Diseases from Basic Clinical Practice. So the first presenter would be Dr. Agyan Jevilano Barinda, PhD. He finished his PhD from Kobe University Graduate School of Medicine, and he graduated before from the faculty of Diponegoro Medical Faculty of Diponegoro University. He's currently the staff lecturer of Pharmacology and Therapeutics Department, FKUI, as well as the PI of Molecular Aging Division in the Metabolic Disorder Cardiovascular Aging Cluster. He already performed many interesting research on aging and also metabolic diseases. And Dr. Jeffy will present about the impact of vascular aging on the metabolic disorder. Please, Dr. Jeffy, time is yours.
Okay, thank you very much, Dr. Diki. Good morning. Assalamualaikum warahmatullahi wabarakatuh. So I would like to share my screen. Okay, is that my, can I show the slide?
Yeah, it's good, Dr. Jeffy.
Okay, thank you very much. Good morning. Assalamualaikum warahmatullahi wabarakatuh. So my name is Jeffy from Metabolic Disorder, Cardiovascular and Aging Cluster. So I just took a photo a couple of weeks ago of the name of our cluster here. So I'm very excited to discuss about the relation between the vascular aging and also the metabolic disorder, focus on the endothelial senescence. Why? Because some people might ask me, what is the relation between the metabolic, cardiovascular, and also aging? So at this moment, I would like to discuss about those relations. So before I start the discussions, I would like to give some important keywords here. The senescence, it means the growing old. So the senescence can determine the aging at cellular level. So this is my outline. So yeah, before we discuss about the endothelial senescence, we would like to know first, what is the function of the endothelial functions in each specific organ? We know that the endothelial cells plays an important role in angiogenesis. So there are two important papers which published by the Dr. Shimizu and also my previous laboratory. So the Dr. Shimizu and his friends found that the reduction of the angiogenesis in the heart can induce the cardiac dysfunctions. And in my previous laboratory, we found that the increasing of the angiogenesis in adipose tissue can convert the white adipose tissue into the brown adipose tissue and further improve the insulin sensitivity. So that's the important role of the endothelial cells and also the angiogenesis in heart and also in the adipose tissue. So how about the function of the senescent endothelial cell? So before that, I would like to describe the type of the senescent cells. The first type is the replicative senescence, which represented the chronological aging in humans, like 1 year old, 7 years old, 20 and 70 years old. The second one, the most important ones, is the premature aging. That I can show here that the pre-adipocyte isolated from the obese patients at 20 years old can be detected the aging marker here. As we can see, the bluish color here in the pre-adipocytes. It means that the premature aging or premature senescence at cellular level can be act as the bridging between the risk factor and also the disease. Okay, so we will talk about the endothelial cells accumulation in chronological aging. As we can see here in this paper, that in both of the veins and also arteries in the aged people, we can detect the endothelial senescent in these vasculars. And how about in the premature aging? So this is the initial and the famous paper about the impact or the accumulation of the endothelial senescent in human atherosclerosis, which published by the Professor Minamino, the famous person in vascular aging field. So they found that the coronary artery derived from the atherosclerosis patient, they can induce the aging marker here, as we can see the bluish color here, but not in the internal mammary artery. So it means that the endothelial cell senescent can be accumulated in both of the replicative senescent, but also in the premature senescence. Okay, so after we know that the senescent endothelial cell can be accumulated in the aging, so how about the quantity and also the quality of the endothelial cell senescence? So if we are talking about the quantity, so we know that the capillary fractions happens in the aging, for example, in the kidney and also in the brain. And interestingly, Dr. Shimizu and Dr. Yoshida, which will present at this workshops, have shown that the capillary fraction also happen in the age phase, as we can see here. The age phase has a reduction of the vascular or capillary fractions, and it can be detected by the reduction of the VEGF-A, and further induce the widening of the brown adipose tissue and leading to the systemic metabolic dysfunction. So at this moment, we can conclude that by increasing the VEGF can also inducing the angiogenesis and improve the systemic metabolic dysfunction. However, there is a pro and also the contra about the impact of the VEGF in age-related metabolic dysfunctions. For example, here, this paper showed that the VEGF is not essential for improving the age-related insulin sensitivity. As we can see here, that actually in the mice at the 60 months old, maybe around the 70 years old in humans, that the vascular phenotype is not rather decreased, but is maybe similar with the young age, but and also maybe increased than the young age. And how about the VEGF level itself in the adipose tissue? So we can see here that the VEGF level is rather increased in the adipose tissue in the age mice. And we can see here also that instead of introducing the VEGF, inhibiting the VEGF can rather improve the metabolic dysfunction here, as we can see here. And also the inhibition of the VEGF can improve the insulin sensitivity, not only in the young mice, but also in the old mice. So this is the first paper talking about the contradiction of the VEGF. However, recent papers which published in Science this July this year show that the VEGF is rather beneficial for improving the age-related insulin sensitivity. As you can see here, that after VEGF administration, there improvement of the metabolic function, as you can see here, that the increasing of the thermogenesis, the reduction of the fat, and also can extend the health span and also the lifespan. As you can see here, the vascular rarefaction happens in both of the brown adipose tissue and also the white adipose tissue in old mice. But after VEGF administrations, of course, the vascular is rather increased in this adipose tissue. And what is the impact? The impact is the browning of the white adipose tissue is happen after VEGF administrations. And VEGF administration can rather extend the lifespan of these old mice. However, we should remember that the VEGF is costly associated with the cancer progressions. Many papers already discussed that the VEGF can induce the cancer progression. Even in this paper, saw that VEGF administration is rather delay the cancer progression, not induce the cancer progression. So this is the controversy paper. And what is the conclusion? The conclusion may be the quality may rather important than the quantity itself in endothelial senescent, as the Dr. William Osler mentioned that a man is as old as his arteries. However, the question is how to measure the quality of the senescent endothelial cell in vivo, not in the cell culture system. The next question is how to identify the direct impact of the senescent endothelial cell only on age-related metabolic disease in vivo. So to answer the first questions, so we generated the endothelial cell-specific senescent mice, which means the senescence process occurs in endothelial cells only. So like in this movie, this mice is getting older, especially in the endothelial cell when these mice are still young. So we generated two types of these mice by using the TRF2 mutation. How? Because the TRF2 mutation can induce the chromosome end-to-end fusion and rather induce the senescent cells. So what is the consequence? So we found after we confirmed or validated the senescent phenotype in this endothelial cell, we found that the senescent endothelial cell induce several pro-inflammatory factors. And interestingly, we can see here that the IL-1 alpha is the highest of the pro-inflammatory factors instead of other pro-inflammatory factors. And then we presume that the IL-1 alpha maybe orchestrates the SASP or the group of the pro-inflammatory factors derived from the senescent endothelial cells. Then we knocking down the IL-1 alpha and saw that the IL-1 alpha knocking down is not only reduce the IL-1 alpha level, but also other pro-inflammatory factors. So what is the relation between the IL-1 alpha and also the metabolic disease? So interestingly, another paper saw that the IL-1 alpha inhibition or IL-1 alpha knocking out in the mice can improve the metabolic dysfunctions after high-fat diet administration. So at this moment, we concluded that the IL-1 alpha can orchestrate the pro-inflammatory groups and then further induce the metabolic dysfunction in the mice. So what is the impact? At the normal diet feeding in our mice, we found that in the 20 weeks olds that the mice can spontaneously induce the insulin resistance. 20 weeks is rather like 17 years old in the humans. And then in the 50 weeks olds, so we found that these mice can spontaneously induce the age-related cardiac failure and also the renal failure. 50 weeks old is like a middle age in the humans. Okay, and at this moment, at the molecular level, if we know that the IRS1 and also the AKT level is important in the insulin sensitivity molecular signaling, and we found that the IRS1 and also AKT is rather reducing in the endothelial cells and specific mice. And how about in the 50 weeks olds? So as we can see here, that the mice become a geriatric phenotype like a kyphosis, loss of the hair, and we found that the cardiomyocyte size is rather greater or increase in the our TG mice, and also the glomerulotic index is rather increased, indicated the age-related renal failure in our mice. So the question is why? Why the endothelial cell senescence can induce the metabolic dysfunction, even if we are giving the normal diet, not the high-fat diet. So the first reason is the endothelial senescence secrete the SASP in the mature adipocyte, and after that, the adipocytes can increase the oxidative stress in this adipocyte, represented here. And after we giving the antioxidant and acetylcysteine, we found that not only the oxidative stress can be inhibited, but also the senescent or the aging in adipocyte can be inhibited also. And what is the consequence? The consequence is the antioxidants administration can improve the metabolic regulation after SASP induction by the endothelial cell senescence. Okay, so this is the first evidence. How about another evidence? What if we generate or someone else generate the endothelial cell specific anti-aging mass? So what is that? So we know that the endothelial cell senescence can be induced by the high glucose conditions, as you can see here. So Dr. Shimizu and his groups, including Dr. Yoshida, which will present also today, generated the endothelial cell specific P53 knocking out mice. What is that? What is the P53? P53 is a guardian of the genome that a balance between the cancer and also the aging. It means if these knockout mice feeding with the high fat and high sucrose diets, this mice is rather getting younger when these mice become older. So in this paper, interestingly, they found that the endothelial cell specific anti-aging mice can reduce the fat size and also can improve the insulin sensitivity, can we see here. And how about another consequence? The enos, which regulates the vasodilator in the vascular, is rather improved in the endothelial cell specific anti-aging mice. Okay, so after we can both of the papers, what is the future perspective of senescent endothelial cell and its impacts? So our group also saw that the senescent endothelial cells not only accelerates the metabolic dysfunction in vivo, but also accelerates the atherosclerosis, the pulmonary fibrosis, and also the pulmonary hypertension. And how about the future treatment of senescent endothelial cell? How about the senolytic, the drug that can directly eliminate or kills the senescent cells only, but not the other healthy cells? How about the xenomorphic? Instead of the killing the senescent cell, this xenomorphic is minimize the impact induced by the senescent cells. And how about the next generation therapies for the cardiovascular and metabolic disorders related with the endothelial cell senescent? So the future treatment and also the next generation therapies will be deeply discussed and presented by the Dr. Shimizu later. So this is my last slide. Thank you very much for your attention. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you very much, Dr. Jeffy, for a very insightful lectures on senescence and vascular aging. I hope there would be a lot of questions. So if you have any questions, dear attendees, you can write down your questions on the question and answer box. You can write either in Bahasa Indonesia or in English language, please. Okay, thank you, Dr. Jeffy. And now we move forward. We change a bit the gear to a bit different topics. It's about gut ecosystem and cardiometabolic diseases, which will be delivered by Dr. Farid Kurniawan. He is a clinician, internal medicine specialist, who is currently doing his PhD in Leiden University Medical Center. He finishes his medical degree and internal medicine specialist both from FKUI, and he's currently doing extensive research on the impact of urbanizations on insulin resistant, especially focusing on the changes in gut ecosystem. He's also currently the vice chair of the MVA cluster as well. Okay, Dr. Farid from Leiden. I suppose this is very early morning in Leiden. I hope you already fresh enough to give a lecture in this early morning. Okay, please, time is yours, Dr. Farid.
Yeah, thank you, Dr. Diki, and for the nice introduction. And good morning, everyone, ladies and gentlemen. Today, I would like to share you a little bit about the topic of gut ecosystem and cardiometabolic diseases. Let me share my screen. Yeah, is it already in the slideshow mode?
Not yet.
Yeah.
Yeah, good.
Okay. Yeah, good morning. Assalamualaikum warahmatullahi wabarakatuh. Like I have told you before, today I would like to share you a little bit about the gut ecosystem and cardiometabolic diseases. But due to limited amount of time, I would like to focus more on the type 2 diabetes, as this one is a very nice representative of cardiometabolic disease. And as we all know that type 2 diabetes is a cardiovascular risk equivalence. So it would be interesting to see the relationship between gut ecosystem and also the type 2 diabetes. And also later on, we will talk about how this relationship could interpret to the therapeutic opportunity or potential. So in Indonesia right now, we are undergoing epidemiological transition in the type of diseases when the prevalence of non-communicable diseases is increasing, like Dr. Diki already mentioned before, obesity, type 2 diabetes, and also cardiovascular diseases. But on the other hand, communicable diseases like malaria, tuberculosis, dengue, and helminth infection or parasitic infection are still quite high prevalence, especially in some part of Indonesia. And in Indonesia now, there is a rapid socioeconomic development, and this rapid socioeconomic development are increasing the urbanization rate. And this urbanization are believed, and many research have shown that really affect these two major health problems in Indonesia. And the definition of urbanization, not only the migration of people from rural to urban, but also the urbanized rural area, the rural area that really highly adopt the urban lifestyle. As we all know that urban is characterized with a westernized diet, with a high fat, high calorie diet, fat dense food, and more sedentary lifestyle with pollution and also higher stress level. Meanwhile, in rural, there is a lower calories and fat intake with more traditional food, and they are more physically active, and there are more pathogen exposure and farm environment exposures. These very big differences between urban and rural environment are believed to cause the differences in the prevalence, either the non-communicable diseases or communicable diseases between these two populations. These are the proposed scheme that this urbanization or the difference of urban and rural could influence our metabolic homeostasis, like the diet and physical activity, also the endocrine disruptors and the pathogen exposures such as helminth infection could influence the energy balance or energy homeostasis in our body, and it could influence the immune system and the inflammation. But not only influence directly the immune system and inflammations, this these changes between two populations could also influence our gut ecosystem. And this changes in the gut ecosystem and the immune system could eventually have effect in our metabolic homeostasis and could make an insulin-resistant condition, obesity, type 2 diabetes, and also cardiovascular diseases. As I already mentioned regarding gut ecosystem, there's a question. So what is gut ecosystem? Many research or many studies use the microbiome as a reference for gut ecosystem, but for our talk today, I would like to include one of the part of gut ecosystem, although not found in every people, this microorganism, which is here will be focused more on the parasites, the soil transmitted helminths. And the microbiome itself is considered as the microorganism, consists of the gut microbiota, not only the bacteria, but also the fungi, virus, and also protozoa. And its gut metabolites and also the gut permeability. This slide shows us the amazingness of this gut microbiota. I believe that gut creates something not without purpose, and we can see here that this gut microbiota could outnumber our own human cells by 10 times, and 95% of this microbiota located in our intestine or gastrointestinal tract. So there is a very a lot of studies show the importance of this gut microbiota in the adiposity and also the immune system. One research by Ridaura and team shows, this is very known research, that the fecal microbiota from 4 human female twin pairs that discordant for obesity. So the one female twin, the BMI is less than 25, and the other twin BMI is more than 30. And this fecal mass are transplanted or transferred to germ-free mice, so they have no microbiota. And the recipient mice that got the microbiota from the obese team, they develop increased adiposity or become obese. Meanwhile, the mice that receive the microbiota or the fecal mass from the lean twin, they develop a lean phenotype. And interestingly, if these two mice are co-housing or cohabited, then the obese mice become leaner. So there is a exchange of microbiota. This experiment or this study is the first study that showed the causality of this gut microbiota towards adiposity. And if there is something that changed this balance of the gut microbiota, or we call it dysbiosis, that can be caused by diet, other age, lifestyle, genetics, and antibiotic use, it could potentially cause certain metabolic diseases like obesity and type 2 diabetes because of certain way, like because of the function of the host gene expression, energy harvesting, and also chronic inflammation. We in the cluster, Dr. Diki, Dr. Dante, and I now performing a pilot study to see the profile of the gut microbiome in the dysglycemia spectrum from normoglycemic, pre-diabetes, and type 2 diabetes or over diabetes. We can see here that we confirm from previous study, diabetes subject is associated with lower diversity of the gut microbiota. Although we did not see a significant difference in the pre-diabetes condition compared to normoglycemic, but from this PCA analysis, we can see that there is a different gut microbiota composition between these 3 groups. Like here, the green one is more on the pre-diabetes, and the blue one are the normoglycemic, and the red one is the diabetes. Although it's not entirely separated, but we can see there is a skew that goes into different direction between these 3 groups. And also, if we look further in the abundance of the phylum, we can see that here the Bacteroidetes phylums is decreased in diabetes compared to normoglycemic, and also the ratio of firmicutes and Bacteroidetes is increasing in diabetes. If we look into further to a lot of previous study, there are a lot of shift of gut microbiota composition, especially in the genus levels in type 2 diabetes. We can see here that several genus like Proseburia, Bifidobacterium, Fecalibacterium, and Akkermansia are decreased in the type 2 diabetes patients. Meanwhile, this genus is increased in type 2 diabetes. although from one study to another study, a lot of differences that could be observed. Now we move on the role of the intestinal permeability in type 2 diabetes. Like I've shown you before, that gut microbiota also composed of our intestinal barrier, that the surface area is more than 4,000 meters square, or is similar with 2 tennis court. But the other composition of the intestinal barrier is also the physical barrier that consists of vascular endothelium, epithelial cells, and mucus layer, the chemical barrier, the digestive and the antimicrobial, the digestive secretion, and also antimicrobial peptides, and also the intestinal immune system. Dysfunction of gut barrier is to allow this absorption of the nutrients in the diet, and also to prevent the entry of antigens and microorganisms into the body. If by some means there is something happen in this intestinal barrier surface, then it could cause the chance to get like nutrient malabsorptions, autoimmunity, food sensitivity, and also metabolic dysfunction. Here we got one examples that high fat diet are that really known as its effect on the increasing intestinal permeability by modulating the gut microbiota composition and also reduction of the tight junction proteins. The increase of intestinal permeability will cause the metabolic enzyctosemia condition. This metabolic enzyctosemia condition is due to the elevation of the circulating lipopolysaccharides that escape into the systemic circulations, and they activate the toll-like receptors on the immune cells. And this activation of the immune cells will cause the condition low-grade chronic inflammations, with associated in insulin resistance. And in the long term, it could cause the condition of type 2 diabetes. There's a lot of ways to measure this intestinal permeability. One way is dual sugar absorption test, and also another biomarkers for intestinal integrity, like intestinal fatty acid binding protein, zonulin, calprotectin, or lipocalin 2. Several studies have shown that there is a higher intestinal permeability in type 2 diabetes compared to healthy subjects, like here by Cox and colleague. They found a higher lipopolysaccharide binding protein and also intestinal fatty acid binding protein in type 2 diabetes, and here also for zonulin. Now we move on to the gut microbiome metabolites and this association with metabolic diseases. Many studies have shown that the effect of gut microbiome are mediated by secretion of various metabolites, and the very important metabolites is the short chain fatty acids. It consists of acetate, butyrate, propionate, and also succinate. This short chain fatty acid is the product of fermentation of dietary fibers and also resistant starch by the gut microbiota itself. And its function is very critical for the intestinal homeostasis, for energy metabolism, and also for immune response regulations. One of the most important for this short chain fatty acid is the butyrate. A lot of studies have shown that there is a decrease of butyrate producing bacteria, like I've mentioned before, the Roseburia, Faecalibacterium, and Bifidobacterium in type 2 diabetes. And butyrate also plays essential roles in maintaining the intestinal integrity. Our cluster team also have shown the impact of sodium butyrate in the inflammatory conditions, although they did not found the difference of butyrate levels between normal glycemic and poorly controlled type 2 diabetes, but they found, or they observed, that this butyrate significantly reduced pro-inflammatory cytokine production in LPS-stimulated PBMC cultures, which is more pronounced in normal glycemic subjects compared to type 2 diabetes subjects. And after I talk about the microorganism or the microbiome part of this gut ecosystem. Now we move on to the macroorganism. Here, the nice examples is the soil-transmitted helminth infection. Our colleagues, Dr. Wirya, have shown by study in Nangapanda, Flores, East Nusa Tenggara, that people with STA infection is associated with lower insulin resistance that's independent of its effect on the BMI. And we can see here, it seems that there is dose relationship between this helminth infection with the insulin resistance, which can see more helminth infection or higher species number of helminth infection are associated with lower insulin resistance. And how do the relationship between helminth infection and the effect on the metabolic diseases? Here we can see that helminth infection very closely related with the adiposity and also the effect of immune system. The helminth and helminth-derived molecules from a lot of studies have shown to skew the immune system toward anti-inflammatory condition. Here we can see an alternatively activated macrophage or M2 macrophage, and also type 2 immune response that consists of Th2 and regulatory T cells. These alternatively anti-inflammatory conditions are associated with less inflammatory condition in our body, and so less insulin resistance. So this also showed the relationship between the insulin resistance, the immune system, and also helminth. And further study that performed by our moderator today, Dr. Diki, in Nangapanda, also the sugar spin study, they performed a cluster randomized double-blind placebo-controlled trial that administered albendazole, an anti-helminth, for 1 year with 4 episodes in almost 1,700 subjects, and they showed that the warming caused the increase in insulin resistance. So this is the first study that showed the causality between helminth infection and also insulin resistance. Another study, they also found that the removal of helminths also increased the pro-inflammatory adipokines, in this case leptin and leptin adiponectin ratio, also the decrease of the anti-inflammatory adipokines adiponectin. We all know that these inflammatory adipokines are really associated with the insulin resistant condition in human body. So there is questions then. So how is the interaction between gut microbiome and solid transmitted helminth? Yes, there is a really close connection and have been shown by a lot of studies between this gut microbiome and solid transmitted helminth. And this interaction could modulate immune response in human. And how helminth affect gut microbiome? It's by its antimicrobial activity, the alteration of nutrients and the niche itself, the changes in the mucus dynamic, and also the modification of immune response by innate or adaptive. And we all know that the path for the pathogenesis of type 2 diabetes, we previously heard the triumvirate, the ominous octet, and now the latest one is the gregarious 11. And one of them are the colon or microbiome. So after we talk the association between this gut microbiome or gut ecosystem with the type 2 diabetes, now we come up to the potential of treatment for type 2 diabetes that related with this gut ecosystem. We can see here that a lot of glucose lowering drugs that we know very effective and fastly used in type 2 diabetes also actually affect the gut microbiota itself. Here, the example is metformin, the first line drug for type 2 diabetes. They changed the Akkermansia, increased the proportion of lactic acid bacteria, and they also alter bile acid metabolism, increase levels of short chain fatty acid, and improve intestinal permeability. Also, we found that DPP4 inhibitor changed the Bacteroides phylum and Bacteroides genus abundance, and they can also increase the short chain fatty acid, particularly succinate. And interestingly, GLP-1 receptor agonists, although it's not administered orally, they administered by injection, they could also change the microbiota composition and alters lipid and sulfur metabolism. And how about pre or probiotics? Is there any effect on type 2 diabetes? Several meta-analyses have shown the benefit of pre or probiotics on type 2 diabetes, like shown by Tao and colleagues here, that probiotics could decrease the A1C, fasting blood glucose, and also the insulin resistance. But it's important to note that this meta-analysis come from the studies that are small in number of subjects, so limited number of participants, and there is significant heterogeneity in terms of the design, the doses of the probiotics or prebiotics, the durations, and also the composition of the bacteria itself. And there is a modest improvement in this lot of studies, with more pronounced in certain group of subjects. So it's not applied for all subjects. So as to summarize, here we can agree that gut ecosystem plays important role in the pathophysiology of metabolic disease, particularly type 2 diabetes. And this we know this association are as another potential modalities to treat or even to prevent type 2 diabetes, although more research are needed to establish the optimum possible intervention toward the type 2 diabetes, and also we could say more on the personalized medicine. Thank you for your kind attention, and would like to receive or give any comment or questions. Thank you.
Thank you, Dr. Farid, for a very interesting lecture on the role of a gut ecosystem, from the mechanism and as well as the potential interventions to halt or alter the pathogenesis of type 2 diabetes, insulin resistance, and also in inflammations. If all the attendees have questions, please write down your question on the question and answer boxes. I hope there would be many questions because very interesting topics. And then we move forward to the next session, see about the third speakers, Associate Professor Shimizu. He finished Chiba University Faculty of Medicine, as well as the doctoral course from the Chiba University Graduate School of Medicine. He's currently Associate Professor, Department of Cardiovascular Biology and Medicine, the Centennial University Graduate School of Medicine, member of AMED and project for elucidating and controlling mechanism of aging and longevity from Japan Agency for Medical Research and Development. Thank you very much for coming here to give a lecture on our session, Professor Shimizu. Time is yours.
Okay, hi everybody. So thank you very much for kind introduction. I would like to thank JFE and the organizer for this giving me this great opportunity to share our published as well as unpublished data. So hope you see all what I see. Okay, so I would like to start now. Okay, so as JFE kindly introduced most of our published papers, so we are interested in the aging process for example, the aging process in adipose tissue or endothelial cells or cardiac tissue, and the close connection with obesity, diabetes, and heart failure. And also, of course, the aging itself. So these are almost like most of them are published papers, so I will skip and just show the scheme. So these through these findings, we take two strategies at this moment. So suppose the young cells become aged, so they are molecules that promote senescence. So we define them as senocules, like senescence plus molecules. So one strategy would be to suppress senocules. And another strategy would be specifically deplete senescent cells, like described as senolysis by Kirkland in United States. So through these strategies, so we can rejuvenate and even reverse the aging process. Okay, so before that, I would like to briefly introduce our published papers. So we are the most of our effort is focused in understanding and trying to generate the next therapies for heart failure. As we know, 1 to 6% of the whole population is suffering from these deadly disorders, and sometimes described as the epidemics. And there's so many unmet medical needs still remains to be explored. So among those patients, these patients are described as non-responders. The prognosis is unacceptably low. So these patients, heart failure patients, are taking the optimized therapy, including like ICD, but still, the prognosis is very poor, as you can see here. So we analyze like extracted cardiac tissue biopsy samples, and then analyze idiopathic dilated cardiomyopathy patients, IDCM patients, and then in the remote phase, classify them as responders or non-responders. So we define non-responders as those patients whose ejection fraction improvement is less than 10%. So there are not many clinical difference between responders and non-responders in the baseline. Okay, so the non-responders, the ejection fraction remains low, but the responders, as we defined, make a recovery as this. And the question is, why there is a difference between the responders and non-responders? So we are interested in electron microscopic findings, and then so the case 1 to 5 indicates these patients are from different patients. So the non-responding patients, we found that mitochondrial size would be reduced, and then we quantify this data. So the non-responding patients has smaller size for the mitochondria, but the number of mitochondria seems to be similar between the groups. So we are interested in analyzing the players for mitochondrial biogenesis, like and also for the dynamics. So as you know, the mitochondria make a fission and also make a kind of fusion. So mitofussion 1 and 2 is a critical player for the mitofussion process. So we analyze mitofussion 1, showing a reduction in non-responding patients in protein as well as in the transcript levels. So non-responding patients have less mitofussion 1. And then we did some functional studies with the neonatal rat ventricle myocytes and NRVMs. We knocked down this on mitofussion 1, and then oxygen consumption ratio would be reduced as predicted, and also the mitochondrial red diminished with the mitofussion 1 depletion. And then we generated cardiac specific mitofussion 1 knockout model, and then subjected thoracic aortic constriction, like TAC model, showing the reduction in the fractional shortening, as well as left ventricular dilatation, and also the fibrotic area will be enhanced, and also the depletion of mitofussion 1 in cardiomyocytes subjected to TAC leads to the increase in the dysfunctional mitochondria, as you can see here. And also like a fibrotic markers and also the transcript increase on the cardiomyocytes. This were enhanced in this condition. And we also characterize the microRNA 1405B. So this is the scheme of the findings. So in non-responding patients, this microRNA would increase via adrenergic receptor signaling, and then microRNA 1405B reduce mitofussion expression, and then mitochondrial respiration is reduced in non-responding patients. So enhancing mitofussion 1 or suppression of this microRNA may become a next generation therapy for the challenge case for heart failure. Okay, so as an introduction, I just wanted to say that considering about the metabolites or system metabolism is critically important to understand heart failure and of course aging. So this process is still mysterious. But in my opinion, the most important mysterious aspects of the aging is why they're going to make kind of a synchronization. Some of them are highly proliferated, like bone marrow cells. Some of them are terminally differentiated, like cardiac tissue, cardiomyocytes, or brains, or whatever. But as we know, we have a kind of a concept that in parallel with aging, they show kind of a functional decline. So I think this is really mysterious. So through the studying the electron microscope findings, I now consider that the metabolite in circulation or protein in circulation has a causal role for the synchronization of aging. So I classify them as senocules. Like I said, molecule that show senescence, and these senocules will have a role for synchronization of aging, sync aging. So far we identify xenometabolite or xenoprotein. Okay, so I brought this figure from the review article. Now we are in revision actually. So there are several molecules that would increase or reduce with aging. So these are characterized in humans. Okay, so Dr. Yoko Yoshida, who is sitting back to me, she will talk in detail about TML, but we are interested in trimethylamine N oxide, TMLO, in heart failure. So TML destroys mitochondria and has a causal role for promoting pathologies in heart failure. So she will talk about in detail for this mechanistic point. So we recently identified that the TML would increase in mice in circulation, and also in the circulation of humans with aging, as you can see here. So we are now collaborating with Tohoku University, who has a metabolized data for more than 2,000 individuals. So it clearly indicates that with aging, the TML level will increase. The point is that what will be the connection with aging? Okay, so as I have already shown you that with aging, TML would increase in mice, and then we generate TML fusion model, and then these mice, the running distance would reduce in the treadmill examination test, as you can see here. And also when we analyze the skeletal muscle, so we found that the more robust phenotype compared with the cardiac tissue, so they show kind of a ballooning phenotype with this crystal in skeletal muscle. Okay, so with the TML, it sounds clear to us that they promote some pathologies in heart failure and also with sarcopenia. And the question would be whether there will be any role for other disorders. So this is an interesting paper published from some other groups. So they're analyzing Alzheimer's disease patients. So as you know, mitochondrial dysfunction has a role for Alzheimer's disease. And then when we analyze like cerebral spinal fluid, they analyze that the TML level will increase in the CSF of the Alzheimer's disease patients. And also another group analyzed the Parkinson's disease patients, and also in the cerebral spinal fluid, as well as in plasma. And the TML will increase in the Parkinson's disease. So what I'm now considering and trying to establish the concept of metabolic related disease, SRDs. So we know that the TML will increase in circulation and also destroys like mitochondria. And the mitochondrial disease will cover like Alzheimer's disease, Parkinson's disease, sarcopenia, and heart failure. So these will be a kind of a syndrome. So that's our opinion. And now I'm trying to establish a kind of research consortium to test this concept, like the SRDs. So there are several other candidates. Sorry, I cannot disclose at this moment, but in addition to TML, there are several metabolites that would increase with circulation with aging in humans. So the question will be whether they will become a therapeutic target. Okay, so that's a talk about xenometabolite, and I would like to talk and share the xenoprotein study. Okay, so this started from a very simple question. So for the visceral fat, they secrete like TNF, interleukin 6, and MCP1. And the question is that whether brown adipose tissue also secretes like functional brown adipocoins. So through studying two data sets of microRNA data for the vascular endothelial growth factor A knockout mice in adipose tissue, including brown adipose tissue, also high fat, high sucrose diet fed mice. We analyzed that the common pathogenic molecules, whose molecules the function is not clear. And then, sorry, I cannot disclose the name for the patent issue, but I'm interested in in one molecule. Today, I will describe the obesity-associated profibrotic protein. So this OFP will increase with obesity in brown adipose tissue and also in the liver. So this is transcript data. And in protein data, there's a robust enhancement in the brown adipose tissue with dietary obesity. Okay, the point is that this is a secreted type molecule. So we found that the OFP will increase in circulation in the dietary obesity condition. And then the point, the keyword would be like obesity, fibrosis, and maybe like unmet medical needs. So there will be like a non-alcoholic steatohepatitis, NASH. So you generated systemic as well as a brown adipose tissue specific knockout model. And then this shows in decline, as you can see here. And when we analyze the liver, the picrocytic staining positive area will diminish if we deplete them in the brown adipose tissue. So this is a scheme of our ongoing project. We generated 3 knockdown models: brown, systemic, and also peptide fractionation therapy. They contribute for reducing the fibrosis in the liver. Also, the gain of function model are showing the enhancement of the fibrosis in liver. And the point is that even in the NASH patients, this molecule will increase. So we consider that this has a potential to become the druggable target for NASH. So we recently identified that with aging, this molecule will increase in circulation in humans, that already reached statistical significance. And also in the mice with aging, they will increase in circulation in protein level, and also in the brown adipose tissue in transcript level. So we are still working on showing the underlying mechanism for this. Importantly, I made a kind of a consortium for this. So in the control group, the OFP level will be like this. In the heart failure preserved ejection fraction, atrial fibrillation patients, CKD patients, non-alcoholic steatohepatitis patients, sarcopenia patients, they have a high expression profile for this pro-fibrotic protein circulation. So what I am now thinking is that with aging and obesity, there are several disease that develop in the heart, like HFpEF, and also for atrial fibrillation, maybe like lung fibrosis, sarcopenia, myelofibrosis, chronic kidney disease, and even NASH. So these may be in general considered as an independent disease, but there will be like a large clinical trial showing a close connection in between, for example, NAFLD or NASH or HFpEF. So what I'm thinking is that these should be described as kind of one syndrome. So I'm now trying to establish a concept like age-related fibrotic disorders, AFib, including these disorders. And OFP will become a kind of like a biomarker as well as a druggable target. So I'm now trying to establish a therapy for targeting OFP and for the AFib. For this aim, I'm now trying to establish a research consortium who has an expertise in the biobank and also clinical studies, genome studies, of course, very extensively do the studies, including the development of neutralizing antibody. And I was lucky enough to win the National Academy of Medicine in USA like last year for this AFib research consortium study. Okay, so using the rest of the time, I would like a little bit make a dive into the brown adipose tissue. So why I and others, our group interested in brown adipose tissue. So this is a white adipocyte. So there's a clear difference between brown adipocyte, as you know. So they are enriched in mitochondria, like cardiomyocytes, and they are initially characterized as a cell and a tissue important for generation of the heat. But nowadays, as I already may have shown, it is highlighted and focused as a tissue or cell that is critical role for metabolic regulation. So in 2009, is a landmark paper showing that with aging, the brown adipose tissue will decline, as you can see here, and also with an increase in BMI, the brown adipose tissue will decline, as you can see here. And also recently, there's a paper published in Nature Medicine showing that again with aging, the brown adipose tissue prevalence will decline, as you can see here. And also non-detectable brown adipose tissue has a close connection with type 2 diabetes, dyslipidemia, coronary artery disease, and even heart failure and hypertension, several vascular disorders. And also, the point would be whether there will be a kind of safe way to enhance the activity of the brown adipose tissue. And we are now interested in the coagulation factors. So as you know, the coagulation factor 10A would promote the production of thrombin, fibrillin, and this is critically important for clot formation, as you know. But our study indicates these coagulation factors promote brown adipose tissue dysfunction. So we analyzed the microRNA data and found that in the brown adipose tissue, there are some transcripts related with the coagulation factors like factor 7, 10, or protease activity receptor 1 would increase in the brown adipose tissue, not in liver. So we found that the transcript factor 10 would increase, and activated form factor 10A would increase in the brown adipose tissue, in protein, also in the transcript. And we found that the protease activity receptor 1 is predominantly expressed in brown adipose tissue that would increase with dietary obesity, with some blood, and also the immunofluorescence studies support these findings. And we introduced factor 10 inhibitor rivaroxaban and found that in the obesity they show like a whitened phenotype in the interscapula brown adipose tissue that show the reduction. So it's kind of a rebrowning phenotype thanks to the factor 10 inhibitors. Also in the cold tolerance test, the thermogenic response will be enhanced, and also they have glucose intolerance will improve in the dietary obese mice subjected with administered with a factor 10 inhibitors, as you can see here. So we did some kind of cold exposure in the obese mice. So the red signal is it's close to the brown adipose tissue. They are existing in the interscapular area. So in the obese mice, the cold exposure will not enhance the signal. But in the brown adipose tissue power knockout model subject to high fat diet, they have enhanced thermogenic response against cold exposure, as you can see here. So this indicates that suppression of brown adipose tissue power 1 signaling would promote enhance the activity of the brown adipose tissue. So this is the scheme of our ongoing project that are now in revision. So the coagulation factors deriving from liver and also lung, white adipose tissue, or even the brown adipose tissue itself, the TF factor 7A and factor 10A make a kind of a complex and activate power 1 signaling, and then the excessive reactive oxygen species promotes mitochondrial dysfunction and brown adipose tissue dysfunction. Okay, so I hope these unpublished data let you convince the critical role of xenometabolite and also xenoprotein. So these should be classified as xenocues, or maybe other microRNA, or I don't know, some other unknown as well as known compounds could be classified as xenocues, and then has a causal role for synchronization of aging. So think aging is similar or equal to think aging. So in summary, brown adipose tissue regulates systemic metabolism, but dysfunction and disoriented choline metabolism from a pathology in failing heart. And Dr. Yoshida will discuss and show the data in detail afterwards in this session. And in final, xenocues contribute for think aging may become a druggable target. And in conclusion, suppression of xenocues, xenometabolite, and xenoprotein may become therapy for age-related disorders. Thank you very much for your kind attention.
Thank you, Professor Shimizu, for a very novel lectures on xenomolecules, xenoprotein, and so on. It's really mind-blowing. So we would go to the question and answer session. We have already received quite some questions from the audience. So the first questions, maybe this can be answered by both Dr. Jeffy and also Dr. Shimizu. Is there any role of immune senescence in this endothelial aging process? Is it more humoral immune response or cellular immune response? Maybe Dr. Jeffy can first address these questions.
Okay, thank you very much, Dr. Diki. So instead of discussing about the immune senescence, so we should know that all of the cells, when the cells become senescent, it can induce the inflammation, or it's called inflammaging. So both of the immune cell and also the non-immune cell can induce the inflammaging, or can secrete the pro-inflammatory factor called the SISP. Our previous lab already published that the senescent endothelial cells can accelerate the atherosclerosis by recruiting the several immune cells like the macrophages and also the inducing some humoral factor like FICAM or something else. So based on this paper, so yes, it can induce the immunosenescence by due to the endothelial senescence. Or maybe the Dr. Shimizu has the better answer for this. Thank you.
You would like to add something, Dr. Shimizu, on the role of immune senescence on aging?
Yeah, so this is a very interesting, important question, and we don't have published data at this moment, but I didn't have time to show share our research regarding the senolytics. So we are interested in senolytics. So in our opinion, there are two types of senolysis. So one would be, the direct senolysis, so kind of a compound or whatever, we directly kill senescent cells. We also have a project related with indirect senolysis. So some kind of a molecule will be critical, important for the regulation of the activity of the immune cells. So the immune cells will be, like others also are published and also showing some unpublished data. So immune cell itself has a role for clearance of senescent cells, possibly including like senescent end cells. So it's kind of an indirect senolytic. So the senescent immune cells is critically important for the clearance of senescent endothelial cells, but we don't have published data at this moment. So it's a great question, to be honest. Yeah. Thanks.
Okay, thank you, Dr. Shimizu. Dr. Farid, would you like to add something about the role of immune response in terms of metabolic disease? Because you also knew about the meta-inflammation as well, right? So would you like to add some?
Yeah, I think I could not add more to the very detailed explanation of the had already mentioned by Dr. Jeffy and Dr. Shimizu. It's already very clear, yeah.
Okay, thank you. So we move to the next questions for Dr. Farid. Could you please explain the pathophysiology of gut microbiota in obese? Why will it lead to adiposity phenotype? The different adiposity phenotype from Dr. Etika Nur.
Yeah, thank you, Dr. Etika Nur, for the question. It's quite interesting also. So the pathophysiology of the gut microbiome related to obesity is by the increase of the food harvesting. So this composition of the gut microbiome from obese people, they are very effective in the harvesting of the nutrients from the food. They induce the secretion of the hormones or the enzyme from the intestine that could absorb more of the nutrients, and also the fermentation of the disaccharides or the carbohydrates in the food content. So this could increase the adiposity by increasing the nutrient uptake, and also they could also influence the appetite of these obese people, this indirect effect of the gut microbiome on obesity. That's my answer, Dr. Diki.
Okay, thank you. Continue on the microbiota topics. There's a questions from Dr. Indrarti about the role of temulawak and kunyit on the gut microbiota. Have you have any info on that issue?
That's very interesting questions. Honestly, I don't have any data regarding this use of herbal medicine, especially very specific Indonesian herbal medicine, temulawak and the other one that mentioned. So what I know is that I think, if I'm not mistaken, there are several studies regarding the traditional Chinese medicine that could affect the gut microbiome, but I don't know whether it's related directly with this Indonesian herbal medicine. So it would be nice to see this Indonesian herbal medicine towards the effect on gut microbiome. So it will need more research on this type. Yeah.
Okay, yeah, definitely more research, because we don't have any data on the role of Indonesian herbal medicine, not yet, at least until now. Maybe first to Dr. Farid before we move back to Dr. Shimizu and Dr. Jeffy. How about the role of probiotics, Dr. Farid, to prevent insulin resistance and cardiovascular risk among those with obesity?
Yeah, interesting. So I've already mentioned in my presentations that indeed from several meta-analyses that probiotic and prebiotic have roles in the reducing insulin resistance or glucose levels in this obese patient with type 2 diabetes or not obese with type 2 diabetes. But it's worth noting that this study are more, although there are several randomized clinical trial study, but they are more in a small number of participants. And like I said, there's a significant heterogeneity. So we still don't know which subjects that are suitable for this therapy, because the pathogenesis of type 2 diabetes that I shown before is multifactorial. We need a personalized medicine. We still have to know which subjects or which patients that are mostly gonna benefit for this therapy, and what type of bacteria compositions, and how much is the doses, and how long we gonna give this pre or probiotics. So it still needs a lot of research, and although it's part of the potential treatment, but it's might not can be applied for all of the patients. So that's my answer.
Thank you, Dr. Farid. So we will move forward to the next questions about the role of exercise in cellular aging. There's a questions, how can it prevent the cellular aging process? Because exercise could also increase the free radical, right? Maybe we can go first to Dr. Shimizu. Do you want to address these questions about the role of exercise on aging?
Yeah, so aging itself, like similar with the calorie restriction, it's a kind of an anti-aging machinery. So they may increase some loss or whatever, but they're not that high. So I think it's the most efficient way to reverse aging. But we are trying to find a kind of a mimetics for similar with exercise, because many of my patients do not feel like doing exercise. Some of them cannot do physically exercise. So we would like to have a kind of a mimetics for the exercise. I have a positive opinion for exercise, of course.
Okay, thank you, Dr. Shimizu. Dr. Jeffy, would you like to add something?
Yes, as Dr. Shimizu mentioned before, that the exercise, the calorie restriction is, we call it the metohormesis. Metohormesis is inducing a mild of the oxidative stress. So it's like a vaccine that can, we call it the what doesn't kill you make you stronger, like this one. So if we can induce the mild of the oxidative stress by the calorie restriction or the exercise, surprisingly, it can induce a kind of the anti-aging process like this one. But of course, not the excessive of the exercise, of the excessive of the calorie restriction that can induce the severe oxidative stress. Yeah, something like that.
Okay, thank you. Dr. Farid, would you like to add something about the impact of exercise?
Yeah, I think I agree with Dr. Shimizu and also Dr. Jeffy that like exercise, we all know that it could improve, I think, also the composition between the brown and white adipose tissue. So I don't know whether indirectly also it could improve the aging phenotype of these patients that perform regular exercise. And yeah, for the treatment of obesity, type 2 diabetes, exercise is one of the main modalities for this disease.
Okay, thank you, Dr. Farid. So we go to the next question for Dr. Jeffy from Dr. Sri Suryani Wijaya. how to balance the activity of p53 to maintain the aging system and tumor suppressor activity?
Okay, so this is actually the interesting question, and I think it's better to Dr. Shimizu to answer because Dr. Shimizu already published this article about the endothelial p53 inhibition. So maybe Dr. Shimizu?
Please, Dr. Shimizu.
Yeah, so this is one of the frequent questions regarding p53. You know, it's and also a kind of a very challenging question. So the answer will be, you know, we don't have a direct answer at this moment. But, you know, too high p53 level, that may be not good, and too low is also not good. So it should be maintained on a kind of a physiological level. So most of our mice studies we did in a homozygous depletion. So, for example, homozygous depletion of p53, and then subjected to, like endothelial cells subjected to high-fat diet, the mice had an anti-obese phenotype for several months. But after 6 months of age, they start to die because of the cancer. So, of course, the homozygous depletion is not good because they promote like tumorigenesis. You know, physiologically, we cannot quantify the p53 level. So together with the potential tumorigenesis, now we make a shift to deplete senescent cells, like p53 positive or p16 positive cells should be depleted. So we are not trying to suppress the process of cell senescence itself because of the potential the cancer development. So the depletion of senescent cells, I think it's more safe, and not many potential side effect issue will be raised if we deplete it. And you know, the bystander effect, the senescent cells will promote the aging phenotype in the surrounding young cells. So if we can specifically kill those cells, the tissue and even the mice, they make a kind of a rejuvenation, so we can reverse aging phenotype when we deplete it. So that's our strategy. Yeah, it's a really important question. Also kind of a headache. So I don't have a direct answer for that.
Okay, thank you, Dr. Shimizu, for the very detailed explanations. So
Dr.
I would like to ask about, yeah, sure, Dr. Farid, please.
Yeah, may I ask one question to Dr. Shimizu related with obesity-associated fibrotic protein. So if you mentioned it's related with the fibrotic, especially in the, I think if I'm not mistaken, the liver fibrosis for the NFLD NAFLD, and is there any, maybe you know, data about these roles of protein in the type 2 diabetes, like maybe the fibrosis condition or the degradations of the beta cell functions in the pancreas itself, whether this also associated with type 2 diabetes? Thank you, Dr. Shimizu.
Thank you very much. We don't have the data at this moment, so we roughly characterize the systemic as well as brown adipose tissue knockout mice models, and we don't have any data for pancreas. It's a very small tissue, so yeah, it's a little bit challenging for us. So maybe with the aging, for example, like obesity, they show like a fibrosis in the pancreas. Am I correct?
Yeah, especially in type 2 diabetes also.
Yeah. So yeah. So we generated 2 transgenic models. One is humanized OFP transgenic overexpression model, and one is mice overexpression model, and that works with a Clear-Lock system. So it's we are now have a limited mice colony. So our plan will be for this year and within 2 years to characterize which organs have a fibrotic phenotype in which organs, so including maybe like pancreas. Thank you very much for your kind attention. Kind question. And also we are interested in like bone marrow cells, for example, or lung, or even brain. Maybe, you know, the capillary fibrosis in the brain has a causal role for Alzheimer's disease, for example. So we also have a plan to analyze the Alzheimer's disease model. And then when we inject a neutralizing antibody, now we are currently screening. If they show a kind of improved cognitive function, we can say that fibrosis may be a druggable target for Alzheimer's disease as well.
Yeah, very
It
interesting.
depends on the transgenic mice phenotype, I guess. I believe they have a role. Thank you very much for your great. It's a great suggestion.
Okay, thank you, Dr. Shimizu. I'm also interested about the OAFP. Can you also measure that in human? I mean, whether you have any data on people with obesity in human? I mean, how it relates with the cardiometabolic complications of obesity? Yeah.
So I'm interested in generating a therapy for heart failure with preserved ejection fraction. So SGLT2 inhibitor that was published, in New England Journal of Medicine, was a kind of a promising therapy for HFpEF, but still we just have one bullet to treat, HFpEF patients. So we need more drugs for HFpEF. There are so many drugs for HFrEF, So our data indicates with aging, they would increase in humans, and also in the HFpEF patients, they show an increase. Also in the NASH patients, there are increase, and also atrial fibrillation patients, they show an increase. So I did for targeting the atrial fibrillation patients in the age-matched patients. So I found that with the age-matched individuals, patients with atrial fibrillation have a higher level for this molecule. So this indicates there is kind of a genetic regulation that makes some specific population to become prone to age-related fibrotic disorders. So our plan is to, I have a collaborator who has more than 2,000 plasma, and also together with the genetic information. So with supported with a bioinformatician and also running the supercomputer, we can analyze whether there will be kind of a SNP that regulates the expression level of this fibrotic protein. So yeah, there's so many things to be done, but undone at this moment. Give me 5 years to answer your questions.
We will look forward for the results. So we fortunately we have already reached to the end of our session. It was very interesting to hear the three of you spoke about the basic science on the obesity, Dr. Farid on the gut ecosystem, as well as how it also may lead to vascular aging, as well as to senescence, also the issue about the new novel issue on the obesity-associated fibrotic-related protein, which is very interesting. And we hope that this meeting not only you could share all your experience, all your research, but also hopefully in the future we can also collaborate on doing some exciting research. I think Dr. Jeffy will then contact you, Dr. Shimizu, to lead the collaboration forward.
Yeah, I'm happy to collaborate. Let's make a kind of consortium all together. Yeah,
Yes.
It's my great honor.
Yeah. Okay, once again, thank you very much for all the speakers, Dr. Shimizu, Dr. Farid, also Dr. Jeffy. We already reached the end of our session. And now I would like to give back the floor to Ms. Tika as the Master of Ceremony. Once again, thank you very much.
Thank you very much.
Thank you, Dr. Diki, Dr. Shimizu, Dr. Jeffy.
Dr. Jeffy, Dr. Farid.
Thank you very much.
Thank you, Dr. Diki Tahapari, Dr. Aikian Jefilano Barinda, Dr. Farid Kurniawan, and Ipei Shimizu Sensei for the interesting presentation. Excellencies, participants, ladies and gentlemen, we are now continuing our meeting to the second session, obesity and cardiovascular, a complex interplay. Okay. This session will be moderated by Dr. Luciani, specialist of internal medicine, consultant of cardiovascular. She completed her medical studies program from MD Pursuit Specialist at the Faculty of Medicine of Universitas of Indonesia, and now she is a staff and lecturer at the Division Cardiology, Department of Internal Medicine, Faculty of Medicine, Universitas Indonesia, Dr. Cipto Mangunkusumo National Referral Hospital, and also as principal investigator at the Cluster of Metabolic Disorder, Cardiovascular, and Aging IMERI FRE. Please welcome Dr. Luciani.
Thank you very much, Ms. Tika. Good morning, everybody, ladies and gentlemen, dear colleagues, professor, doctor, fellow students, and all participants. Assalamualaikum warahmatullahi wabarakatuh. Salam sejahtera untuk kita semua. Maybe Ms. Tika already introduced me. So yes, my name is Luciani, and today I will be moderating this session. As part of this very wonderful webinar, this session will talk about obesity and cardiovascular as a complex interplay. Today, we will have a distinguished speaker from Japan, Dr. Yoko Yoshida, and also our distinguished speakers from Indonesia, Professor Dr. Sidartawan Sukondo and Dr. Bambang Widiantoro. Good afternoon to Dr. Yoko Yoshida, and good morning to Professor Sidartawan and Dr. Bambang. It's an honor for us here to have you all today. Before we start, let me remind all participants to write down your question in Q&A box, and I will deliver those questions to related speakers in discussion session. So our first speaker is Dr. Yoko Yoshida, who will deliver a lecture with the title of Brown Adipose Tissue-Derived Metabolite as a novel therapeutic target for heart failure. Dr. Yoshida is an associate professor in Juntendo University Graduate School of Medicine, Department of Cardiovascular Biology and Medicine. She was certified board member of the Japanese Society of Internal Medicine in 2008 and Japan Circulation Society in 2013. She went also a fellow of the Japanese Society of Internal Medicine in 2016, and she had a lot of achievement, I think. Including Jokinshi Takamin Memorial Award in 2021 and Young Investigator Award in 2020. Without any further delay, please, Dr. Yoshida, time and screen is yours.
Thank you for your kind introduction, Dr. Luciani. This is my great honor for me to be invited to this great meeting, and I'd like to thank the organizers for giving me a chance to talk today. This is my COI. As you know, an increase in the number of patients with heart failure is an international health-related problem, which is recently called heart failure pandemic. And it is well known that rates of death and disability caused by severe heart failure are still high, which is sometimes higher than some kind of cancers. Therefore, there are unmet medical needs, and it is our urgent task to find new therapies, not only focusing on heart, but also other organs. This previous report shows that low body temperature predicted a poor prognosis in heart failure patients. However, the underlying mechanisms and pathological implications are largely unknown. Brown adipocytes are enriched in mitochondria and recognized as heat-generating organ. Brown adipocytes are activated by cold exposure and adrenergic signaling, and fatty acids are utilized as fuel, and heat are generated through UCP1 in mitochondria. And recently, some studies show that brown adipose tissue is present at human adults and has a function for systemic metabolism. And it is also shown that detectable human brown adipose tissue declines as aging, and also shown that obesity caused whitening of brown adipose tissue, leading to an impairment of systemic metabolic homeostasis, such as thermogenesis and glucose metabolism, indicating that brown adipose tissue has crucial roles for the maintenance of systemic metabolism in obesity. However, a link between heart failure and brown adipose tissue has not been fully elucidated. So we aimed to elucidate the role of brown adipose tissue in the pathologies of heart failure. For this purpose, we generated heart failure model by thoracic aortic constriction TAC operation. As you can see, cardiac function deteriorates in 4 weeks together with the hypothermia and impaired thermogenic response in cold tolerance test. In this condition, brown adipose tissue became reduced in volume and hypoxic. And we also found reduced UCP1 expression and an increase of apoptosis in brown adipose tissue in TAC model. So we hypothesized that pressure overload promoted brown adipose tissue dysfunction. It has been reported that sustained sympathetic nervous system activation promotes cardiac dysfunction. And we found that the norepinephrine level in brown adipose tissue was significantly increased during pressure overload. And continuous infusion of isoproterenol impairs thermogenic response together with an increase of apoptosis or in brown adipose tissue. We also found that treatment of cultured brown adipocytes with no epinephrine impaired mitochondrial function and increased apoptotic cells, suggesting that activation of adrenergic signaling suppresses mitochondrial function in brown adipocytes. Next, consistent with this data, surgical denervation of sympathetic nerves innervating into brown adipose tissue, which effectively reduced brown adipose tissue norepinephrine level, improved cardiac dysfunction after TAC operation. Next, we performed TAC operation in genetic brown adipose tissue dysfunction mice. Brown adipose tissue specific mitofusion 1 and 2 double knockout mice. As you know, mitofusion 1 and 2 are essential for maintaining the mitochondrial homeostasis. So depletion of mitofusion N2 leads to mitochondrial dysfunction and consequently whitening and dysfunction of brown adipose tissue. And as you can see, brown adipose tissue dysfunction significantly worsened the mortality and cardiac dysfunction after TAC operation. Similarly, we performed TAC operation in combination with brown adipose tissue removal. After BAT removal, thermogenic response was reduced in CTT, cold tolerance test. In this model, we found that cardiac dysfunction and fibrosis were reduced, were significantly worsened in brown adipose tissue removal group. Next, we transplanted healthy brown adipose tissue into white type mice to enhance the brown adipose tissue function, and then perform TAC operation. In this model, we found that brown adipose tissue transplantation ameliorated thermogenic response and improved cardiac dysfunction during pressure overload. These data indicate that during pressure overload, excessive adrenergic signaling promotes apoptosis of brown adipocytes and brown adipose tissue dysfunction, thereby inducing the progression of heart failure. Then, to clarify the mechanisms in which brown adipose tissue dysfunction promotes heart failure, we analyze the changes of metabolites in heart failure model using mass spectrometry through collaboration with Professor Soga in Keio University, and found the increase of choline in brown adipose tissue and its metabolites trimethylamine N oxide TMAO in plasma and heart in TAC model. And in genetic and surgical brown adipose tissue dysfunction model, we found an additional increase of TMAO. And interestingly, choline was reduced in brown adipose tissue transplantation model, suggesting that choline is metabolized in healthy brown adipose tissue. In the previous reports from Norway group, circulating choline and TMAO were increased in human heart failure patients. And more importantly, their level were much more advanced in heart failure patients whose NYHA class was 3 or 4, or heart failure is coronary artery diseases. So we decided to focus on choline and TMAO in this study. Then we performed TAC operation followed by TMAO infusion. In this model, we confirmed an increase of TMAO model and found that a TMAO worsened cardiac dysfunction and fibrosis. Similarly, TAC model with high choline diet whose TMAO level was higher than normal level also showed worse cardiac dysfunction and fibrosis. Next, we tested the effect of inhibition of choline oxidizing enzyme COE on the progression of heart failure. We performed TAC operation followed by treatment with COE inhibitor via drinking water. And in this model, we confirmed an effective reduction of TMAO production and found that cardiac dysfunction was significantly improved by COE inhibitor. Similarly, genetic depletion of COE by COE knockout mice also improved cardiac dysfunction and fibrosis. And electron microscope findings clearly show the disruption of mitochondrial crystal structure in hearts after TAC with TMAO treatment. And the metabolomic analysis showed the decrease of ATP production and phosphocreatine level after TMAO treatment. And extracellular flux analysis using isolated mitochondria of TMAO-treated hearts show the reduction of mitochondrial complex 4 function. To further investigate the detailed mechanism, we performed comprehensive proteomic analysis using isolated cardiac mitochondria from TMAO-treated mice. As you know, there are complex 1 to 5 in the mitochondrial oxidative phosphorylation pathway, and complex 4 is composed of some cytochrome C oxidase such as COX-1. And in our proteomic analysis, we found that COX-1 was decreased by TMAO treatment. And we confirmed the reduction of COX-1 by Western blot analysis. And it is also known that HEM-A is essential for the assembly of COX-1 into complex 4. Then we found a decrease of HEM-A and its precursor HEM-B in TMAO-treated mitochondria. So we speculated that TMAO would inhibit the enzymatic activity of ferrochelatase, FECH, which catalyzes the production of HEM from protoporphyrin 9 with ferric ion. And we found that administration of TMAO suppressed FECH-induced metabolism of protoporphyrin 9, suggesting that TMAO inhibits the production of HEM and destabilizes COX-1. Finally, we analyzed the human heart failure patients in our hospital and found that choline and TMAO increased in heart failure patients together with hypothermia. We also checked the brown adipose tissue activity in heart failure patients using PET-CT. Among 2,989 patients who was examined by PET-CT in a hospital since 2015 to 2019, 309 patients were enrolled in this analysis. Exclusion criteria was described here, and we found that brown adipose tissue tends to decrease in heart failure patients. These data suggest that brown adipose tissue dysfunction would be important for the progression of heart failure in humans. In summary, during pressure overload, excessive adrenergic signaling induces brown adipose tissue dysfunction, which leads to the metabolic disorientation and accumulation of choline. Accumulating choline leaks to the systemic circulation and is metabolized to TMAO, which promotes cardiac metabolic remodeling. In conclusion, suppression of TMAO and maintenance of brown adipose tissue homeostasis would become a novel therapeutic target for heart failure. I would like to thank my mentors, Professor Minamino and Dr. Shimizu, and also my colleagues in Minamino Lab and all of collaborators and foundings. Thank you for your attention.
Thank you very much, Dr. Yoshida. It's very interesting presentation. I'm sure a lot of questions from the audience, and we will discuss it later, okay? And then we will proceed to our second lecture from Professor Dr. Siddhartawan Sugondo, who will talk about obesity and atherogenic dyslipidemia future direction. Professor Siddhartawan is one of our honored professor in Faculty of Medicine, University of Indonesia. Professor Siddhartawan had a lot of experience and also in educational field, as he went for in endocrinology in University of Amsterdam, University, and also several courses in diabetes from Singapore, Germany, Denmark, Swiss, San Diego, and USA. So without any further delay, please, Professor Dr. Siddhartawan Sugondo, time and screen is yours.
Okay. Thank you, Dr. Luciani. It's now my turn. Let me share my screen. Okay, once again, thank you very much. Assalamualaikum warahmatullahi wabarakatuh. Today, my lecture is more clinical. Let me see. Yeah. Okay. My talk today is about obesity, atherogenic dyslipidemia actually, but about the future directions. As we all know that obesity will reduce our life expectancy, will impair our quality of life, and will cause disabilities. And mainly will cause also cardiovascular disease, type 2 diabetes, osteoarthritis, and cancer. Actually, obesity is associated with multiple complications. On the metabolic part, you see here that we can get non-alcoholic fatty liver disease, gallstones, infertility, and cardiovascular diseases, stroke, dyslipidemia, hypertension, coronary disease, pulmonary embolism, and HFpEF. Type 2 diabetes, pre-diabetes, thrombosis, gout, cancer, and besides metabolic disorders, mechanical, they cause asthma, incontinence, GERD, knee osteoarthritis, sleep apnea, and chronic back pain, and of course also depression and anxiety. Obesity is actually meets the common criteria of a disease. According to American Medical Association, it should have an impairment of the normal function of some aspects of the body, characteristic signs of symptoms, harm, and morbidity. The impairment is appetite dysregulation, abnormal energy balance, endocrine dysfunction, infertility, novel dyslipidemia. And the signs and symptoms are increased body fat, symptoms associated with increased body fat, including joint pain, immobility, and sleep apnea. And of course, it causes harm or morbidity, and it causes type 2 diabetes, cardiovascular disease, cancer, osteoporosis, and polycystic ovary syndrome. Malnutrition, sedentary lifestyle, genetics, social, cultural, and economic conditions will cause obesity, insulin resistance, inflammation, and of course, with epigenetics, metabolomics, and proteomics will cause metabolic syndrome and diabetes. And we all know that metabolic syndrome and diabetes will cause atherogenic dyslipidemia. This will be then the link between obesity and atherogenic dyslipidemia, where there will be an increase in triglycerides, lipoprotein, increase in HDL cholesterol, and increase in small dense, low density lipoprotein, and it cause atherosclerotic cardiovascular disease. If we look forward, visceral obesity leads to insulin resistance and reduced glucose availability. And of course, after that, the fat stores use energy, release free fatty acids because they are not energy enough, and there will be free fatty acids will increase hepatic uptake of fatty acids, and then will increase hepatic triglycerides production in very low density lipoprotein and will cause small dense LDL and small dense HDL. Now, the atherogenic lipoproteins, as we all know that very low density lipoproteins, intermediate and density lipoproteins, low density lipoproteins, LPL3. And these are the atherogenic lipoprotein classes. Why atherogenic dyslipidemia? Because atherogenic dyslipidemia contributes to excess risk of cardiovascular disease. The higher the triglycerides, the lower the HDL. You see here that the odds ratio for coronary heart disease is higher. Thus, there is a correlation between the elevated TGs and low levels of HDL cholesterol have a synergistic detrimental impact on residual cardiovascular disease risk in patients on target LDL cholesterol. Yes, if the patients are already on target treatment, there is still a residual risk of these patients. Yeah. Now, and the current risk management, LDL cholesterol is currently the primary target for dyslipidemia management, but patients treated with medications targeting LDL cholesterol, for example, statins, still have residual cardiovascular risk. For this reason, other targets are being considered, including non-HDL cholesterol. This is what I'm going to talk about, beyond LDL cholesterol. We all know that triglycerides is definitely a marker of risk before, but now there's a possibility that it's going to be a marker of an underlying disease. Despite benefits of statin-induced LDL cholesterol lowering, treated individuals have substantial residual risk for coronary artery disease events. All these trials will show you that the relative risk reduction, there's still residual risk after treatment with statins for LDL cholesterol lowering. Thus, we have now to take care of, to look forward with an emergent treatment target, the non-HDL cholesterol. We know all that HDL cholesterol is anti-atherogenic, and the atherogenic lipoproteins are LDL cholesterol, IDL, and VLDL. Yeah, non-HDL cholesterol may be a better marker of cardiovascular disease risk than LDL cholesterol in patients with high TGs and diabetes, metabolic syndrome, or chronic kidney disease. According to the European Society of Cardiology, the guidelines said non-HDL cholesterol should be considered as a secondary target, and the maximum recommended level is LDL cholesterol goal plus 30 milligrams per deciliter. And to calculate this non-HDL is very easy. Non-HDL is total cholesterol minus HDL cholesterol. Why is it important? Why should we treat non-HDL cholesterol? If you see here, there are 2 patient cases with same total cholesterol, 209 milligrams per deciliter. Without calculating the non-HDL cholesterol, case 2 might not be treated and therefore may be at risk, since non-HDL cholesterol goal should not exceed 30. If you see here that non-cholesterol here, total cholesterol is 209, yeah, and then the LDL cholesterol is 89. If you only look at the LDL cholesterol, then we're not going to treat it because LDL is quite normal. But if you see here, the total cholesterol is 209, but the triglycerides are about 100. And the non-HDL, if you have 209, you minus HDL, the non-HDL will be 177. While actually it should be the LDL goal plus 30. At the most, it should be 130. But if you see the case number 1, the LDL is 145. You treat it. But if you see the second patient is 89, and you are not going to treat it. But the non-HDL is 177. This patient has a high non-HDL and non-HDL goal. The non-HDL cholesterol includes an assessment of all ApoB containing lipoproteins, considered to be atherogenic, all ApoB. Yeah? The good correlation with ApoB is VLDL, IDL, LDL, and even Lp(a). The non-HDL is an indirect estimate of LDL particle number, and LDL particle number relates more closely to the CVD risk than LDL cholesterol. Non-HDL cholesterol makes no assumption about the relationship between VLDL cholesterol and TGs. In type 2 diabetes, this relationship can be altered, leading to the falsely low LDL cholesterol levels, as I mentioned before, as calculated by the Friedewald formula. And inaccuracy can happen with the Friedewald because also if the LDL cholesterol is less than 70. The practical advantage is that does not require fasting, this is non-LDL, and easily calculated and inexpensive versus ApoB, which is almost equivalent, and can be used in patients with TG levels more than 400 milligrams per deciliter. Thus, the ESC and EAS guidelines for non-HDL cholesterol, the recommendation for type 2 diabetes and insulin resistance, it mentioned that atherogenic dyslipidemia is a major risk factor for cardiovascular disease in type 2 diabetes patients, people with abdominal obesity, and insulin resistance or impaired glucose tolerance. And these patients may derive clinical benefits from TG lowering therapy as an add-on to statin. But of course, the secondary objective for non-HDL, if the patient is high risk, non-HDL should be less than 100, very high risk less than 85. As if you say ApoB, then high risk is less than 80 and very high risk less than 65. The short summary. Dyslipidemia in metabolic syndrome and type 2 diabetes patients. Dyslipidemia represents a cluster of lipid and lipoprotein abnormalities, including elevation of both fasting and postprandial TGs, ApoB, small dense LDL, and low HDL cholesterol and ApoA levels. Non-HDL cholesterol or ApoB are good markers of triglyceride-rich lipoproteins and its remnants, and are a secondary objective of therapy. This atherogenic dyslipidemia is one of the major risk factors of CVD in people with type 2 diabetes and in people with abdominal obesity and insulin resistance or impaired glucose tolerance. After this, there are 2 new papers. The first is triglyceride-rich lipoproteins and their remnants, the metabolic enzymes, the role in atherosclerotic cardiovascular disease, and emergent therapeutic strategies. This is a consensus statement from the European Atherosclerosis Society 2021. It said that recent advances in human genetics, together with a large body of epidemiologic, preclinical, and clinical trial results, provide strong support for a causal association between triglycerides, triglyceride-rich lipoproteins, triglyceride-rich remnants, and the increased risk of myocardial infarction, ischemic stroke, and aortic valve stenosis. These data also indicate that triglyceride-rich lipoproteins and their remnants may contribute significantly to residual cardiovascular risks in patients on optimized low-density lipoprotein therapy. This, if the patient is already treated and the targets are achieved, there is still residual risk. This statement critically appraises current understanding of the structure, the function, and metabolism of triglyceride-rich lipoproteins and their pathophysiological role in atherosclerotic cardiovascular disease. If you see, this is the metabolic pathway of lipoproteins. You see, from the gut, they will have chylomicrons, and from the liver, we have very low density lipoprotein, and then the chylomicrons and VLDL with lipoprotein lipase will have free fatty acids, and will goes to the skeletal muscle, heart muscles, and adipose tissues for energy and storage. And the VLDL itself will then change with HDL, and then they will transfer cholesterol and triglycerides from VLDL, IDL, and LDL. And LDL will cells and vascular walls and will go to also to other tissues. In detail, it's been like this: the overview of probably. There's adipose tissues. Free fatty acids go to the liver, and with lipogenesis, de novo lipogenesis, and B100 synthesis, will cause then will have then VLDL2 and VLDL1, and then this will then after that, because we have VLDL remnants and IDL. This after fairly low density lipoprotein, the VLDL1 and 2 will have two tracks. There will be VLDL remnants and there will IDL, and IDL will become LDL, and both of them will go by the LDL receptor and will go back to the liver. And yeah, and if dietary fat, we have chylomicron, and the triglycerides and cholesterol will then transferred to the skeletal muscle, adipose tissues, and heart muscles. Yeah. Now, if there is diabetes or insulin resistance, then the VLDL overproduction, the formation of triglyceride-rich lipoprotein remnants, and their role in atherogenesis. If the VLDL is normal, free fatty acids are normal, then there will be no overproduction. But if there is diabetes, insulin resistance, there will be VLDL overproduction. Then you see here that the newly secreted particles become transient remnants, become large VLDL, small VLDL, IDL, and also the remnants from the chylomicron. Yeah, they have chylomicron remnants and they have VLDL remnants. The remnant lipoproteins will clear by the liver or will penetrate the arterial wall. Yeah, this will cause small dense LDL. There will be two pathways to become atherosclerosis through the small dense LDL and through the remnant lipoproteins which penetrate the arterial wall. If you see this, the role of triglyceride-rich lipoproteins and remnants in atherosclerosis. The initiation of early focal lesions, the chylomicron remnants, the VLDL remnants, and IDL. Yeah, you see here, because of these remnants, will go through the endothelial cells with transcystosis, yeah, go to intima, and here you see that the difference is between the LDL should, small dense LDL, if it goes to the arterial wall, should be oxidized first. But this is not, it's going to take straight. Yeah, this you see here, the blue one, cholesterol ester from the chylomicron remnant, the VLDL remnants and IDL will go straight transcystosis to the intima, and then there will be a process for the endothelial dysfunction, release of lipolytic products, and the monocyte macrophage recruitment, and then becomes the home cell. And then it progresses to the position of cholesterol, release of pro-inflammatory products, and you see here becomes the formation of fatty streak, and so on. Yeah. Now, if you see the metabolism of the remnant proteins, If the TG triglycerides is less than 100 milligram per deciliter, and compared with moderate to severe elevated plasma triglycerides between 150 to 500. This group is the VLDL overproduction, the increase in liver fat, insulin resistance, and type 2 diabetes. This is the normal production of normal liver fat. Normally, yeah, you see if the lipidation and the remodeling because by the lipolysis, high LPL, low ApoC3, yeah. And then here you see the remnant population is just not many. The branch of the VLDL process leading to remnant formation and clearance and conversion to LDL. Some becomes LDL and there's an end product, the remnant population. But if there is an overproduction, because there are reduced lipolysis, the lower lipoprotein lipase due to genetic variants to high ApoC3, then you see here that there were a larger VLDL pool with increased proportion shunted to remnant pathway. You see the remnant population will be more. The prolonged residence in circulation, remodeling of the core, and surface composition will increase atherogenicity. That will be then in the future to be proven. Yeah, besides the increased small dense LDL. If you compare the remnant particles and the normal LDL, you see here that the remnant particles, because of the accumulation of cholesterol ester via cholesterol ester transfer proteins from the HDL, yeah, you see it here, the cholesterol is more, 2,500 to 10,000 millimole, yeah, and the TG is reduced, yeah. And here you see the decreased particle lipid fluidity, decreased particle net charge, yeah, and then you see the difference between the normal LDL and the remnant particles. So, according to this paper, they categorize the triglycerides. Yeah, just optimally, this should be around 100 or less than 100. The borderline is around between 100 and 150. Moderately elevated is 100 to 500, and severe between 500 and to 880, and extreme more than that. Yeah, more than 880. Dear colleagues, chylomicrons and VLDL, or triglyceride lipoproteins, are assembled in the small intestines and in the liver, and transport TG and cholesterol into the circulation. Sorry. Plasma levels of triglyceride lipoproteins are determined by the rate of production, the efficiency of removal of TG contents by lipolysis, and hepatic clearance. Several enzymes, lipid transfer proteins, and the lipid and protein composition of the triglyceride-rich lipoproteins and remnants all contribute to the regulation of lipolysis and hepatic clearance of these lipoproteins. In severe hypertriglycemia, a reduced TRR clearance rate is the dominant metabolic abnormality, and partial removal of TG from chylomicrons or VLDL creates remnants. And these remnants levels depends on the extent of lipolysis, conversion of VLDL remnants to LDL, and the efficiency of hepatic removal of both VLDL and chylomicron remnants. Let's talk about the journey of this ASC/AIS guideline. Yeah, 2019 ASC/AIS guidelines for dyslipidemia management recognize that the ASCVD risk is increased at TG levels above 150 milligram per deciliter, but only recommend initiating pharmacotherapy in high-risk patients if more than 200, after excluding secondary causes. There are also no treatment goals in these guidelines. Either the plasma TGs or indices of remnant abundance, given limited evidence from randomized controlled trials that lowering TG or TRR reduce ASCVD risk. Just because of this are no goals, there are less trials. This, what is I going to do? Strategic approaches to lowering TRR warrant needed discussion. The point is especially pertinent given that the study of PISA reported the presence of peripheral atherosclerotic plaques in 58% of middle-aged individuals with elevated TGs more than 150, low to moderate cardiovascular risk, and high or normal LDL cholesterol. About clearance, the journey is set at the production and then the clearance. Increasing the efficiency of removal pathways for atherogenic lipoprotein should theoretically reduce ISFD risk. Statins will regulate the LDL receptor and would be expected to decrease remnant particles by accelerating their catabolism. And statin-mediated TG lowering is limited at an optimal levels of TG, but more effective at higher risk, more than 175 milligram deciliter, with similar percentage reduction in TG and LDL cholesterol. The higher the TG, the better the statin will respond. Yeah, the statins also promote high omicron remnants clearance and reduce lipemia following a fat-rich meal. Thus, statins can reduce remnant abundance, providing further support for optimizing their use for individuals most at risk. The conclusion of from the AIS in 2021 is for the triglycerides, lipoprotein, and the remnants is the knowledge of TG metabolism and the aberrations leading to elevated plasma TG levels is substantial. However, understanding the pathobiological of remnant lipoproteins, as I mentioned before, the atherogenic potential of their lipodomic and proteomic composition, and their measurement is very much a work in progress. This is still in progress. That is the key to the development of optimal targets which is going to come. The statement highlights two important unmet needs. A standard readily applicable assay to measure remnants is not in clinical practice yet. Therapeutic options to lower circulating levels of remnants to reduce residual ASCVD risk in patients on maximal LDL cholesterol lowering treatment. With several promising candidates, the answer will likely involve combination therapy targeting both remnant formation and clearance pathways and avoiding attendant increases in LDL particles. And the later key is that because any pro-atherogenic effects of TRR or triglyceride lipoproteins and remnants will be synergistic to those of other ApoB-containing lipoproteins, particularly LDL lipoproteins. Next will be the expert consensus decision pathway of the management of ASCVD risk reduction in patients with persistent hypertriglyceridemia from the American College of Cardiology. Yeah. Now, these consensus recommendations are provided for clinicians and patients regarding unique aspects of lifestyle interventions from management of hypertriglyceridemia and the use of statins and triglyceride risk-based non-statin therapies for ISFD risk reduction in the following patient groups with persistent hypertriglyceridemia. There are 4 groups: patients with ISFD, patients with severe hypertriglyceridemia, patients with diabetes mellitus and additional risk factors, and high-risk primary prevention. You see here, this patient population, you have this patient population, yeah, and then these are the steps, common steps. Factor to consider: persistent fasting hyperglycemia, and then what further to do. The patient with ISFD is adults with ISFD and fasting TG more than 150 or non-fasting more than 175 and below 500. Patients with diabetes is more than 40 years, no ISFD, fasting TG more than 150, non-fasting 175, and TG less than 500. The high risk: adults aged more than 20 years with no ISFD or diabetes, fasting TG more than 150, non-fasting more than 175, and TG less than 500. Patients with severe hypertriglyceridemia, adults more than 20 years with TG more than 500. The steps are the same. Yeah, but if you see each step have the following details. Patients with ISFD has different, yeah, you see here, if persistent high TG on 550 to 499, then there's because this is special, adults with ICVD. After that, they consider the LDL cholesterol guidance non-statin therapy according to the 2018 ACC Multi-Society Cholesterol Guidelines. For the patient with diabetes, yeah, at the end we consider icosapentaen, yeah, omega-3 repair, and then for high-risk patients, yeah, high-risk primary prevention, there's also a guideline special, and also for patients with severe hypertrophy, this is the special group because this is morely not for ICVD, this is more for prevention of pancreatitis. Then all these has different same common guidelines, recommendations for lifestyle intervention in patients with increasing levels of weight loss, and yeah, the difference is between the diet of TG less than 500 to 999 and more than 1000. Yeah, this is for the lifestyle. And then if you see here that the management of ICVD risk reduction in patients with persistent hypertension, but provides comprehensive practical information for clinicians. Yeah, this they mentioned the triglycerides and this guidelines, how to measure triglycerides, evaluation and management of secondary causes of hypertriglyceridemia, lifestyle intervention for management of hypertriglyceridemia, the role of statin therapy in patients with persistent hypertriglyceridemia, and the role for triglyceride-based therapies for ICVD risk reduction. Yeah, this, ladies and gentlemen, today my presentation is about obesity and atherogenic dyslipidemia, the future directions which I presented to take further actions. The work is still in progress. Thank you very much for your attention.
Thank you very much, Professor Sudartawan Sugondo, for your very interesting speech. We will have very interesting discussion later. Maybe a lot of questions to address to you. Okay, and then we'll proceed to our last speaker. Our last speaker is Dr. Bambang Widyantoro, who will deliver a lecture with the title of Obesity and Endothelial Dysfunction, from Basic to Clinical Implication. Dr. Bambang is the Principal Investigator of Cardiovascular Division in Metabolic Disorder, Cardiovascular and Aging Cluster IMERI. He is now the staff and lecturer as well as intensive care medical staff and cardiovascular emergency in Department of Cardiology, Vascular Medicine, Faculty of Medicine, University of Indonesia. Without any further delay, please Dr. Bambang Widyantoro, time and screen is yours.
Thank you very much, Dr. Lucy. Very good afternoon, all my dear colleagues, and warm regards to my teacher, Professor Sudartawan. I enjoy your lecture so much. I would like to share my screen. Okay, can you see my screen?
Yes, we can see.
Okay, thank you very much. So I would like to share with all of you how obesity may cause endothelial dysfunction and how it further caused the cardiovascular disease. I have nothing to disclose related to this presentation. However, I have to admit that I am 170 centimeter height and 85 kilograms. So my BMI by today is 29.4. So Dr. Lucy, I do have a conflict of interest to the topic. Okay, so this is my outline, starting with the epidemiological data and then how the crosstalk of adipose tissue and endothelial cells contribute to the endothelial cells damage in obesity, and further go to the cardiovascular disease. This data from Endocrine Society showing that 1 out of 3 adults in United States have the obesity with BMI over 30 kilogram per meter square, and it more the prevalence is higher even in the women as compared to in the man. However, it's not only, the burden is not only in the adult, but also in the children. So 1 of 5 children start to have overweight or obesity when they start primary school, and may rise to 1 out of 3 when they start secondary school. The data from US, but I think almost similar trend we also may observe in our country currently. So the excess body weight, as shown by this report, is one of the burden of cardiovascular disease attributable to major risk factors, along with the hypertension, diabetes mellitus, and also tobacco smoking. And this is the data from our basic health survey or RISCSDAS, showing that in a decade, the prevalence of obesity is increased twofold, from 11% in 2007 up to 22% in 2018. So, and also the overweight population is also increased. And across 34 provinces, we can appreciate that the lowest prevalence of obesity is in East Nusa Tenggara. I think Dr. Diki, Dr. Farid, and all colleagues from endocrine has a lot of population study there in Nakapanda. And the highest prevalence is in North Sulawesi. And our previous data showing from the population screening for high blood pressure, we can see that the increase of body mass index is very well correlate with the increase of of systolic and also diastolic blood pressure. And this is in adult population over 18 years old. So what kind of clinical phenotype we may face in the obesity, starting with the childhood, and then the pathological clinical condition may be observed in the adolescent and then in the adult phase, starting from sleep apnea, the psychological factors, high blood pressure, dyslipidemia, the disorder of the adipocytes, insulin resistance, and also inflammation. And almost all of the changes may initiated by the dysfunction of endothelial cell in the macro and also the microvascular. So when we talk about endothelial cell, we may remember that this might be the biggest endocrine organ, if we may say so, which potentially regulated the vasomotor, myocardial function, and also vascular homeostasis by secreting so many hormone and peptides along the body. And in the healthy endothelium, we can see that most of the healthy endothelial cells will secrete the vasodilatory peptides, and then anti-inflammatory peptides, antioxidant peptides, and it preserves the barrier function in the vasculature and also in the important organs. However, when the endothelial cells disrupted by many diseases such as obesity and also the diabetes, so the dysfunctional endothelium may change the phenotypes and profile, become the homeostasis or the balance between the vasodilator and vasoconstrictor become disrupted. So it will be a highly vasoconstriction profile and also pro-inflammatory, pro-oxidative stress, and also loss of the barrier function that may lead to the other disease progression in every organ. So diabetes and obesity itself may contribute to the impairment of the enos production and the loss of nitric oxide. And then the inflammatory process will start with leukocyte addition, activation of the platelets, the endothelial production decreased, and the increasing of angiotensin, endothelin, and also prostaglandin. So is it solely due to the endothelial cells itself, or is there any organ involved? It seems like the adipose tissue play a very important role. So many scientists proposed that the obese adipose tissue may have a very close crosstalk with endothelial cells. We know that the important endothelial cells lining in the vasculature and also the myocardium, and the interaction may occurs in perivascular adipose tissue and endothelial cells in vasculature, and also the epicardial adipose tissue with the myocardium endothelial cells. And this crosstalk further cause the more severe endothelial dysfunction. This is a very nice data showing that in the obese patient, when they take out the visceral fats and then take out the very small arteries from the visceral fats, they show that the TNF-alpha expression is enhanced in the obese individuals as compared to non-obese individuals. So it may, the TNF-alpha was suggested to be in the cornerstone of the pathophysiology. And it might be true since the classical mechanism is about the pro-inflammatory pathway and also the increase of oxidative stress. However, so many studies in the experimental studies and also in the animal models also showing actually so many pathways involved in the obesity induced endothelial dysfunction. At least the role of the changes in adiponectin and then the insulin resistance and then the enhanced secretion of vasoconstrictor peptides such as endothelin and also renin-angiotensin system activation has a very important role in this complex molecular pathway. So let's continue to how from endothelial dysfunction become the cardiovascular disease. Almost similar to what happened, in the diabetes condition, in obesity also may lead to cardiovascular disease, at least in two big forms. The first one is the acceleration of the atherosclerosis and cause the coronary artery diseases. And the second one is the direct damage from the obesity to the myocardium that caused cardiomyopathy. Both may lead to heart failure. And this is a very simple way to induce obesity in the mice model. We can just try to give the high-fat diet in the normal mice, and we can compare with the standard diet during 10 to 16 weeks, and we may observe the increasing of body weight, the changes in fasting glucose, and also the insulin. So this is a very common model in animal study to make the obesity mice model. And many reports showing that the pathway in the animal model might be due to the myocardial lipotoxicity in obesity, such hypothesis by this group. And it's very interesting that we observe almost similar condition happen in diabetes. So the diabetes mice induced by streptozotocin, who also have an increase of body weight, when we take a look at the heart and look through the electron microscopy, showing many lipid droplets as compared to the control mice that we use, the vascular endothelial cells knockout mice. And how is the phenotype of myocardium in the obese patient? So the Dallas Heart Study is this very elegant study in the States showing that the remodeling of left ventricle occurs in the obese and severely obese individuals, while probably 30 to 40% may still have a normal left ventricular dimension and also function. However, about 30% showing the concentric left ventricular hypertrophy. And the other paper proposed that the obese heart may show the dilatation of the left atrium, the more thick epicardial fats, and about the left ventricle itself, usually still in the normal or preserved ejection fraction. However, the LV strain, if we measure by global longitudinal strain echo, we may already find the dysfunction. And the characteristic is almost similar to the extent of hypertensive heart disease, which is the concentric left ventricular wall. And this is one of our patient, a 40 years old male with obese, with BMI 31, with hypertension and also pre-diabetes, showing the normal resting ECG and normal coronary artery. And from echocardiography, we may appreciate the concentric left ventricular hypertrophy with still very good LV ejection fraction, but have the clinically phenotypes of heart failure with dyspnea on effort, with the paroxysmal nocturnal dyspnea, and needs heart failure treatment. When we check the cardiac MRI, we try to find the myocardial triglyceride content by MRI spectroscopy, and we compare with a non-obese and non-diabetic individual. We observe that this phenotype of obese and pre-diabetes individual showing a higher myocardial triglyceride content from the cardiac MRI. So, my dear colleagues, what about the future direction? European Society of Cardiology just this year published the consensus paper about the endothelial function in cardiovascular medicine. And they suggest that we may have to carefully screen from the beginning for the patient with cardiovascular risk factors, but before they have the cardiovascular disease, and we may need to check the endothelial function by all these modalities that we may have. The easiest way is, I think, to try to do the flow-mediated vasodilation of brachial artery, the FMD techniques, with all the downfall, I think still remain one of the good examination to show the endothelial dysfunction. And some other propose the finger plethysmography and also retinal endothelial function examination to observe the renal arterioles, and we may need to improve and validate this procedure to be able to observe it in daily practice. So, and how is the current position of the treatment for obesity-induced endothelial dysfunction? So as I presented before, that the crosstalk between endothelial cells and also adipose tissue is very important. So currently, many people try to find out what is the best strategy, whether this is a single target only in endothelial cells or dual target to try to improve both endothelial cells and also the adipose tissue. And as a summary, obesity stimulates endothelial cells damage to very complex molecular pathways. Some hypothesize showing that probably TNF-alpha may be in the cornerstone of pathways. However, the crosstalk between adipocytes and also endothelial cells is very important. And these crosstalks may further lead to cardiovascular disease, including accelerated atherosclerosis and also the direct damage of the myocardium. So any effort in primary prevention in those individuals with risk factors of obesity and also insulin resistance is important to prevent the myocardial remodeling and cardiovascular disease. Thank you very much.
Thank you very much, Dr. Bambang Widianto, for your interesting presentation. So since our speakers have delivered their speech, I would like to invite all participants to have some questions to the speakers. From the Q&A, I only have one question. Maybe the others will proceed. The question is for Dr. Yoshida. How about the effect of exercise on brown adipose tissue? From Dr. Awi Sahab.
Thank you very much for the question. That's a very interesting point, but actually there are some conflicting results in both rodents and humans. Some studies show that there is a beneficial effect of exercise on brown adipose tissue function, but the other shows there is a detrimental effect. Exercise downregulated the brown adipose tissue function. And I myself don't have any data about exercise, but I believe at least the loss of body weight by exercise has a beneficial effect on brown adipose tissue, because we previously showed, actually Dr. Shimizu previously showed that obesity causes brown adipose tissue dysfunction, including thermogenic response and glucose homeostasis. So maybe I believe at least loss of body weight by exercise, it has a beneficial effect. But exercise itself has beneficial or detrimental is unclear yet.
Okay, is it possible, because maybe by exercising there are some reduced inflammatory factors that could about improving the brown adipose tissue, Dr. Yoshida?
Yeah, there is some factors affecting the brown adipose tissue function, such as sympathetic nervous activation and IL-6, as you mentioned, and FGF21 and irisin and so on. So maybe there is complex factors which affect the brown adipose tissue function. So maybe that point make difficult to address to this problem.
Okay, thank you, Dr. Yoshida. Prof. Siddhartha, maybe you have some comments or thought, Prof, on this matter? Okay.
No, thank you.
Okay, okay, and then I have another question, I think. Okay, before we proceed to Prof. Siddhartha, maybe one question to Dr. Yoshida. As you mentioned before, that brown adipose tissue reduced by aging, right? Is there any consideration on or concern if we try to supplement the brown adipose tissue in a geriatric patient or geriatric human being? As we know that geriatric have reduced cardiac adaptation, and the most population of heart failure is geriatric patient.
Yeah, it is. There is a possibility for that, but actually, maybe it may be difficult to improve the brown tissue function itself in aged individuals. So we focused on the downstream molecules of brown tissue that may affect cardiac function or other metabolic condition. For example, TMAO in my project.
Oh, okay. Dr. Bambang, maybe you have some comments?
No.
Okay, thank you, Dr. Yosida. So the research that you have before, this is a suggestion for only for reduced heart failure, or maybe we can. This is a future or alternative therapy for any stage of heart failure. This brown adipose tissue.
Sorry?
Do you suggest for brown adipose tissue is for a future direction or alternative therapy for only for maybe reduce heart failure, or maybe in any stage of heart failure?
We have some data that the brown adipose dysfunction has worsened. Brown adipose dysfunction worsened. Other pathogenic conditions such as glucose metabolism and yeah, so on. So maybe we can expand to other field by moderating the brown tissue function or downstream molecules regarding the brown tissue.
Okay, thank you, Dr. Yosida. So I will proceed to Prof. Sudartawan. There are some questions for you from Dr. Etika Nur. I would like to ask to Professor Sudartawan about the phenomenon metabolically healthy obese, metabolically unhealthy, and normal weight metabolically unhealthy in progression cardiovascular risk.
Okay, thank you. Yeah, actually about metabolically healthy obese, there are many options. I mean, many studies. Some studies say it is as a transition, yeah, but some studies say as equally as non-obese. Yeah, but there are so many definitions, and they are not quite sure about the definition of a metabolically healthy obese. But some say this is still if you are obese, you are a transition to get cardiovascular disease. There's not an agreement that what a metabolically healthy obese is, because of different definitions, and some are saying it's a transition from healthy to unhealthy. Yeah, there's still many studies about it. As far as I know, it's like that. Thank you.
Thank you very much. Thank you very much. And then another question for Siddharthawan. Could it be possible that Indonesian people has slightly different profile of lipid and metabolism and normal levels of lipid parameter related to different body fat size compared to white people? From Dr. Yohanes Purwoto.
Yeah, okay, thank you. If you listen to the first speaker from Professor Senescent, Asian population has different profile. Yeah, East Asian, Southeast Asian has different profile. Yeah, if you talk about East Asians and South Asians, South Asians are more insulin resistant, and the obesity is different fat profile. And also, if you see I mean, the definition of obesity is also a little bit difficult because, I mean, if you use only body mass index or you use waist circumference, yeah, you see, we in Indonesia are not that big as Caucasians, but we still die from cardiovascular disease. We are not that heavy, but we are more visceral obese. Yeah, this, I think we still have to have our own, I mean, our own population study about the fat content. Yeah, because body mass index is mostly for population study. But if you see that obesity is screening, you use body mass index, yeah, body BMI. But to take action, this body mass index has to be with comorbids. Yeah, if there's they have comorbids besides body mass index, then and therefore, as I said before, obesity is to be considered as a disease. Yeah, at least the screening you use body mass index, but to define it as obese, then this body mass index has to a person has a patient with people with obesity has to have comorbids, and if you reduce the weight, then the reduction also for these comorbids. Yeah, that's maybe you compare it healthy obese and non-healthy obese, but there is no such thing as healthy obese because there is still risk. I think this way. Maybe the others can have any comments. Thank you.
Aging also risk, yeah, Prof. Aging also for risk for
Yeah.
atherosclerotic also. So there maybe there are such no healthy person at the end. Because we all have risk for atherosclerotic or cardiovascular diseases. Okay, thank you very much, Professor.
Dr. Bambang can comment healthy obese.
Okay, Dr. Bambang, could you share some comments?
Yeah, I think in the we have all different stages of healthy, right? So what seems to be healthy from the outside, if we look deeply inside, probably not healthy as well. So yeah, one of the very interesting about the obesity is we do have what we call obesity paradox in heart failure patient. So the heart failure patient who become cachexic, the prognosis, the morbidity and mortality is increased as compared to those who can keep the body weight not cachexic. So that's the only quote by quote the advantage of having overweight of obesity. Yeah.
Yeah. Okay. Okay. Thank you very much, Dr. Bambang. So for the next question, Prof. Sunartawan, as you mentioned before, that the TRL level determined by production and then removal and hepatic clearance. Do you have any suggestion how to, maybe you can give us some side of thought, how to manage dyslipidemia, maybe with a high level of TRL in hepatic failure patient, Prof, with known CVD even before, maybe she or he had MCI before, or maybe stroke. It's quite difficult. Sometimes it's quite difficult. Thank you very much.
I cannot answer. I have no experience if you specifically for heart failure patients, because but it's only said that for certain conditions of the heart you need ketones. Yeah, but maybe if you link it to that, maybe it's like that. Yeah, but for this remnants, you see, is because of free fatty acids. Because you saw for diabetes, for insulin resistance, obesity, you saw that insulin, that lipoprotein lipase is controlled by insulin. I mean, insulin is an anti-lipolytic agent. If insulin is not there, then triglycerides will just work very hard, and then your free fatty acids will be more. Yeah, and free fatty acids will overflow to the liver. Yeah, but of course, if it goes to the heart, I cannot answer that. Maybe
Dr. Bambang
because at some stage of myocardial infarction, the myocard uses ketones. Maybe there's some links at that. The ketones is very good for the heart. Okay, maybe link is the cardiologist.
Because we have maybe Dr. Bambang also have some patient with MCI and then cirrhosis, right? Patient with hepatic failure. What difficult to manage dyslipidemia in those patients, Prof? So as you state in your lecture before, there are some beneficial action, I think, if we can control TLR. Triglyceride-rich lipoprotein. Maybe Dr. Bambang can have some comments, Dr. Bambang. Maybe you do have some those kind of patient, right?
Yeah, the dyslipidemic patient is quite common in our coronary artery disease population. However, the specific TLR, I don't have specific experience in following and making the serial measurement with our population, but probably this might be interesting idea for the studies.
So it's quite interesting. Maybe related to your slide also, Dr. Bambang. The patient with dyslipidemia, hypertension, and then you done MRI, CCMR.
And pre-diabetic patient.
Maybe those kind of patient that would give the beneficial of treating more aggressive on triglyceride-rich lipoprotein.
Yeah, I do agree with that. However, the other interesting finding is the high myocardial triglyceride content in our obese pre-diabetic subjects is not linearly correlated with the level of plasma triglyceride.
Oh, I see.
Yeah, so it's very interesting. I will also ask for Professor Sudatawan comments. There's some cases that the plasma triglyceride levels is not so high, probably just between 150 to 180. However, the myocardial triglyceride content we can detect is increased as compared to the non-obese and non-diabetic.
Yeah, I see.
Yeah, can I make a comment?
Yes.
I think we have to differentiate, because the triglycerides or the remnants is actually not actually dangerous for the heart or the vascular, but it's only a surrogate, because it is mostly cholesterol. Why do we reduce triglycerides? Because of pancreatitis, not the effect on the heart or the blood vessels. There's only the cholesterol.
Yeah.
But as you know, the cholesterol has limited. The higher the cholesterol, the better, let's say fibrates will help. Yeah, but the latest study about the reduce it, you reduce the triglycerides with ecosapent ethyl. Yeah, the reduction is there for cardiovascular death, but instead there's a reduction, but the reduction of TGs are not prominent. They said it's not because of the TG. It's only a marker. If you reduce the TG, something, therefore, there's a working in progress that they want to try to see what is the real thing happening. Because if the TG is decreased, LDL decreased, the risk is there, reduced also. But the TG is not significantly low. Something is happening, and that is going to study that. Therefore, he said the end study is still in progress. But by reducing LDL, synergistically with LDL, the reduction will be there. But actually, the one who is responsible is not TG, something else. That's therefore there is a difference, because still TG is only sorry, has nothing to do with vascular. Reducing it is more for pancreatitis. Therefore, it's a secondary target. Yeah, that's what it is. Therefore, we have to separate. Cholesterol is the ones who make atherosclerosis, but triglyceride will cause atherosclerosis also. The remnants, as I said before, yeah, they will go straight. Yeah, the transitosis. Yeah, they will know that the study is that to tell him that because besides LDL, the small dense LDL by diabetes, there's these remnants. Goes also to the transitosis, goes to the intima, and this causes also plaque.
Yeah.
Yeah, there are two things, and this now is studied. Yeah, which one? Because by hepatic lipase, the triglycerides will go to the HDL, but the cholesterol will still be there, and this cholesterol will go this will cause atherosclerosis. I hope you agree. I will just explain. You are both cardiologists. I'm just an endocrinologist. Try to explain more. I think this is the basic.
I have some thoughts there. Okay, thank you very much, Professor Sudatawan. Maybe Dr. Yoshida, you have some comments or thoughts on this matter? Okay, thank you. Okay, and then I will proceed to Dr. Bambang. So this is a question from Dr. Sri Suryani Wijaya. Dr. Bambang, about the endothelial dysfunction. Do you use BWF as a marker? Is it easier to perform in lab?
Okay, thank you for the question. So von Willebrand factors I think it's a good marker. However, if we back into the clinical setting, some large cohort studies abroad show a very weak prognostic properties of this marker. When we apply in the healthy subject, healthy population, and doing the cohort for 20 years, the power to determine the prognostic of cardiovascular disease is very weak when adjusted to the other conventional cardiovascular risk factors. However, we still may have a chance to use this marker in the population who already have an episode of myocardial infarction. So some other cohort studies in the population of recent myocardial infarction showing that the highest von Willebrand factors plasma level may predict the recurrent or reinfarction in the future as compared to when adjusted with other cardiovascular risk. So by those data, I think currently we cannot recommend the plasma von Willebrand factors examination as the screening in the healthy individuals. However, it may have a place for those who already experience myocardial infarction.
Okay, thank you very much, Dr. Bambang. Actually, this is maybe the at the end of this session, but I have one question for Dr. Bambang.
Yes.
Is it okay? Just one question. So, as we know, and also you mentioned before that it's very trending now that we have preschool obese children.
Yeah.
Apparently, also for the past 2 years in this pandemic era. So all children goes to school from home, right?
Yes.
They can do everything from home while eating, just like my son. So my son is getting bigger now.
Yeah,
He's
yeah,
getting
yeah.
bigger now. So do you have any suggestion, or maybe you have some thought on maybe exercising can delay the endothelial function dysfunction that might come to those population? Because I think it's very quite normal to see obese children now since this pandemic era. Thank you, Dr.
Yeah, I think this is a very common phenomenon in this pandemic area. I agree with you. And I don't have any data from my works to answer your questions. However, the effect of exercise to reduce the body weight, I think will have a further beneficial effect to the metabolism of the lipid and glucose, and also further may prevent the endothelial damage. But to what extent the childhood obesity already make the consequences of endothelial damage, I think it still need further studies.
Yeah, I think it's very interesting to study that we can have for
Yeah,
future
I agree.
research. Okay, thank you very much for all the speakers. I think that will be our last question for our session today. I hope everyone will enjoy this session and get enough information from our distinguished speakers. I hope this session will always will improve also our practice in our daily practice to manage our patient. Thank you for Dr. Yoko Yoshida, Professor Sriatawan Sugondo, Dr. Bambang Sugondo, Dr. Bambang Widiantoro for delivering such great lecture and discussion. And maybe from some other time we can meet again and we can do another interesting discussion. Thank you very much. Have a good day.
Okay, thank you.
Thank you.
Thank you.
Assalamualaikum
Thank you,
warahmatullahi
Dr.
Thank you.
Thank you, Dr. Yoshida.
Thank you, Dr.
Thank you, Dr.
Yoshida,
Yoshida,
Dr.
Dr.
Sriatawan,
Luciana, Dr.
Dr.
Bambang.
Bambang.
Nice meeting you all.
Thank you
I
very
hope
much,
to see
Dr.
you again.
Yoshida.
Bye.
Thank you, Dr. Yoshida.
Yeah.
Thank you very much.
I
Bye.
will give the floor to Mystica as the master of ceremony. Thank you.
Thank you for Dr. Luciani, Yoko Yoshida Sensei, Professor Sriatawan Sugondo, and Dr. Bambang Widiantoro for interesting presentation. Excellencies, participants, ladies and gentlemen, we are now continuing our meeting to last session. This session will be moderated by Dr. Farid Kurniawan, specialist of internal medicine. Please welcome Dr. Farid.
Thank you, Mystica, for the introduction. Dear ladies and gentlemen, professor, doctor, colleagues, thank you for still joining us in our masterclass of our cluster, Metabolic and Cardiovascular and Aging here in IMERI FKUI. Now we come to the last session, last but not least for our session today. That will be delivered by our head of cluster, Dr. Diki Tahapari, regarding the prevention of CVD or cardiovascular disease among diabetics, updates from newest guidelines. As already Tika mentioned, Dr. Diki is the head of our cluster, the Metabolic, Cardiovascular and Aging cluster here in IMERI FKUI, and he's also staff in Division of Endocrine, Metabolic and Diabetes in Cipto Mangunkusumo Hospital and FKUI. He's graduated from FKUI and also earned PhD from LUMC. For time and screens is yours, Dr. Diki.
Thank you, Dr. Farid, for the very kind introductions. Good afternoon, professors, seniors, and dear colleagues. So I think my job is quite easy because everything actually has already been discussed before. So for the last 1.5 hours, I deleted almost quite a lot of slides because most of them has been discussed by Dr. Bambang or Prof. Sriatawan Sugondo. So hopefully actually what we're going to discuss in this last session is more about the applications of current guidelines that actually has been presented by Professor Sriatawan, and about the barrier. So I hope we can discuss about how to address these barriers of implementing those guidelines in our patients. So my talk would be more on epidemiological and policy perspective. So I added a bit in the title about the prevention of CVD among diabetics, update from newest guideline and barriers on implementations. So I think you already know that cardiovascular disease remain the most common cause of morbidity and mortality. It's not only in advanced or developing country, but as well as in low and middle income country. And if you look into this graph, you can appreciate that Indonesia has one of the most higher age standardized death rates from CVD, only standing below Pakistan and Philippines. And one of the possible explanation may be, we know previously about the deadly quartet, the dyslipidemia, diabetes, hypertension, and obesity. And if you look into the last basic health research done in 2018, we can appreciate that the prevalence of obesity reaching more than 30%, as previously mentioned by Dr. Bambang. Pre-diabetes also soared to about 30%. Dyslipidemia as well, about 30%. Hypertension as well, more than 30%, as well as metabolic syndrome. The clustering of risk factor have already reached more than 30% of all adult populations. And if you look into the BPJS data, we can appreciate that the cardiac complications has been the top financial burden to the system of the universal health coverage. And this has been also mentioned by Dr. Farid before, that the urbanization is the main driver, which is the socioeconomic development. So the adoptions of urbanized lifestyle, not only those who live in urban area, but also those who live in rural area, has led to the increasing prevalence of obesity, hypertension, diabetes mellitus, and CVD in both area, not only in urban, but as well as in rural area. This is a beautiful study performed in East Java, in Malang, comprising more than 20,000 subjects. You can see here, appreciate the high CVD risk based on the cardiovascular, 10-year cardiovascular risk, reaching about 1/3 of the adult populations. And although the rural area seems a bit lower, if you can see the graph on the left side, actually the difference between urban, rural, and suburban is not that much difference. So it is very important to address this issue in the future. So if you look into the diabetes, you focus on the diabetes, the number of diabetes has also increased rapidly from 5.7% into 10.9% in 2018, which means that about 1 out of 10 Indonesian adults were having diabetes mellitus. And the problem is most of them, more than 80%, were undiagnosed if they were undergone the oral glucose tolerance test. And the most important issue is actually about 1/4 of those people with diabetes in Indonesia are less than 45 years old, which we call so-called the early onset diabetes. And this group of subjects is actually very important because they're prone to develop complications. They're more prone to develop complications compared to those who develop diabetes in the later stage of life. If we look into the complication of diabetes, we can see that the prevalence of cardiac complication reaching about 23% of whole patients with diabetes, although still be lower compared to neuropathy and retinopathy, but the impact of those cardiac complications can be troublesome. This is our latest data from the Bogor cohort study of non-communicable disease risk factors. We try to develop model of which pre-diabetes or diabetes patient will develop cardiovascular event in the next 5 to 6 years. And we can identify there are 3 factors that come up. First, age more than 45 years old. hypertension, and the last one is dyslipidemia. So on those populations, about 82% of those people with pre-diabetes or diabetes had dyslipidemia. And this was associated with increased risk of cardiovascular event by 1.8 times. So I would like to draw your attention to the previous STENO2 trial comparing the conventional therapy versus the intensive therapy. So comparing actually the conventional versus the multiple intervention trial. So the multiple intervention trial will also target not only blood glucose, but as well as lipid, blood pressure, and if needed, antiplatelet, yield a much better outcome in terms of cumulative death, cardiovascular death, death from cardiovascular, stroke, myocardial infection, and so on. You can see on the right side of the slide presentations. And if you try to divide which one contribute more to the risk reductions, you can appreciate that the reduction of lipids contributes more compared to the reduction of A1C or blood pressure. So in treating patients with diabetes, it is of importance to not only treat the blood glucose, but we should also treat the blood pressure, and of most, we need to treat as well as the lipids. This actually has been all described by Professor Siddhartawan. This is the portrait. If we did a population study, which is performed by Professor Swastika in Bali, comparing the prevalence of dyslipidemia on those people with diabetes and normoglycemia. So here you can see the LDL cholesterol, the high LDL cholesterol is affected more than about 80% of the populations. The same goes for non-HDL cholesterol, which is also important, as previously mentioned by Professor Siddhartawan. The ApoB also higher, also the TG also higher. So this is the problem at the community or population level. About 80% of the population had higher level of LDL cholesterol. If we look into the data from Jakarta, which performed about 10 years ago, you can see as well similar patterns. About more than 90% of people with newly diagnosed diabetes had high LDL level. And those with normal even also had about 80% had high LDL. This is also as well translate into other like high cholesterol, high TG, and low HDL. This leads that the problem of dyslipidemia is very huge. And for the past few years, we knew that from the guidelines, we knew that the target is now getting lower and lower. Now the lower is better. I think that's already been addressed before by Professor Siddhartawan. This is because of the data from the 4S study, that the anti-ideal study, IMPROVE-IT study, and that the Fourier study, the use of PCSK9 inhibitor, has shown that the lower the LDL, the better the cardiovascular risk outcome. But then also people look into the residual risks. So although LDL cholesterol is the dominant risk factors, the currently available lipid-lowering therapy are insufficient. In some patients are insufficient to bring halt to the SVD episodes. So people move forward to the non-HDL, which is closely resembles actually the ApoB. So the target now, first the LDL, and the next one would be targeting the non-HDL cholesterol. So there are many guidelines for lipids, but if we want to apply to Indonesia, we need to discuss several things. I think the four most important things to discuss. First is how to screen and assess the lipid disorders. The next one is how to perform the risk stratifications, and then determining the treatment goals, and then treating to target. How can we achieve the target? This, for example, from American College of Endocrinology. For the lab exam for dyslipidemia, we should measure the lipid profile, but also don't forget now to measure or calculate the non-HDL cholesterol. Prof. Siddharthan has already been mentioned before. It's not difficult. You just need to do simple calculations. But we also need to assess other metabolic parameters, for example, A1C, thyroid function, ApoB, or other metabolic complications that might be available on those patients. And if needed, we need to also perform another diagnostic procedures, for example, ECG or CAC scoring, and also carotid intima-medial thickness measurements. And also risk calculators, the risk Framingham score, MESA risk calculator, and so on. So there are several things that we need to perform to clearly stratify whether this person has been grouped as high risk, moderate, or extreme risk. This is one of the examples from the American College of Endocrinology, targeting either LDL cholesterol, first that one, and then non-HDL, and then ApoB. But for limited resources, I think ApoB could be substituted by non-HDL. But also the last one, we need also to target LDL. If you look into this in terms of diabetes, actually it's quite aggressive. If the patient had diabetes, at least we need to target less than 100 of LDL cholesterol. But if it is diabetes with more than one risk factors, one or more, we need to target less than 70. So maybe most of our patients, we need to target less than 70. And if a patient has chronic kidney disease, more than stage 3, or history of premature ICVD, then we need to target less than 55. So for diabetes, at least we target less than 100, and if there is multiple risk factor, less than 70. So I think this is the clinical approach. I tried to summarize in one graph. So we need to stratify whether this is a low risk, medium high, very high, or extreme risk. So if the risk is high, very high, or extreme, we can start directly with the high intensity statin. So if a patient with type 2 diabetes and with one or more risk factors, we can start with high intensity statin. And if needed, we can add, for example, ezetimibe, which is available in Indonesia. And unfortunately, other options were not available in Indonesia. So if you look into the available therapy in Indonesia, we have the statin therapy and also ezetimibe and fenofibrate. The one that is included in the high intensity statin is actually only atorvastatin 40 to 80 milligrams, as well as rosuvastatin 20 to 40 milligrams. I think the data has shown us the data from the atorvastatin, from the Lipitor, showing that the giving of 80 milligrams of atorvastatin could reach the median LDL cholesterol of 57, which is near the target of 55. And it could also reduce the acute cardiac event relative risk reduction of 25%. One of the issue in the diabetes is also most of our patient has already had, as well as a chronic kidney disease. If you look into the effect on the creatinine and albumin creatinine ratio, atorvastatin had a superior compared to the rosuvastatin. So this is the one that is related with hypertriglyceridemia that has already been mentioned by Professor Siddhartawan. I think the one that is quite new, I think the addition of EPA in the guidelines. I would not discuss this further. So this is the new guidelines. So if you look into the guidelines, not necessarily for dyslipidemia, as well as for hypertension, for diabetes, you can see now that the approach is comprehensive. In the diabetes guidelines, you can see they also recommend the treatment for hypertension and dyslipidemia, and the other way around. In the treatment for dyslipidemia here in the ESC 2021, you see not only they also mention about the target for LDL, they also mention about the target of A1C, the blood pressure, and even mentioning the type of drug that can be used for diabetes. This is mentioned as SGLT2 and GLP-1 receptor analog. So what is the difference this guideline compared to the previous one? Because in diabetes, in most cases, we need to do aggressive treatment. In this European Society of Cardiology guidelines, you can see now they have the approach of stepwise. So the first step is actually reduce the LDL, for example, less than 100 or less than 70. It depends whether the patient had established cardiovascular disease or not. And then after reaching this target, then we can intensify reaching the targeted therapy based on the same as the American College of Endocrinology. So the difference only in terms of they provide more comprehensive approach, and the next one is step by step. First, we need to reach certain level, and then if possible, we need to intensify the treatment to reach lower level of LDL cholesterol. But the fact in the application at the community or at the hospital, this is the data from RSCM at 2015, about 6 years ago. The achievement of LDL level less than 100 is only 36%. And if you look at the characteristic for the people diabetic patients in RSCM, actually most of them actually has more than 1 or 2 risk factors. So we need to aim less than 70 or even less than 55. So the one that can reach less than 70 LDL is only about 15%. So it's very low. So how about at the primary healthcare? This is the latest data from Dr. Yunir and Dr. Wafa, studying about around 180 people with diabetes at the primary healthcare. You can see here the number of people with high LDL, more than 100, is about 83%. And the amount of people with dyslipidemia is almost 100%, 96.8% had dyslipidemia. How about the treatment? So is this a treatment? In reality, almost 62% of them did not receive any dyslipidemia treatment. Only about 28% received the treatment, and most of them actually were given a low intensity or even only very small number received at least moderate intensity. So this is the reality in the primary healthcare. Most of them only receive low intensity statin. So I think we come about the last slide of my talk. So this is the repetition of what I mentioned before. To reduce the cardiovascular risk, we need to do comprehensive management. And one of which, the lipids should play also an important role in the diabetes, preventing the CVD. So we need to do collaborations, not only, maybe people come first with the cardiologist or the nephrologist or the endocrinologist, internal medicine or primary health care physicians. We need to collaborate as well with patients, trying to communicate that the target is not only about the blood glucose, but also the lipids and also the blood pressures. So dyslipidemia increases the risk of cardiovascular. The target could be LDL cholesterol, non-HDL, 3G, and also ApoB if available. So we can treat with statin, high intensity in most cases, ezetimibe, fibrate, and also maybe in the next future we will have PCSK9 inhibitor. So this is the available treatment in Indonesia. We have the statins now. We have the high intensity in terms of atorvastatin, also rosuvastatin. We have ezetimibe, we have fibrate. But some of drugs that is mentioned in the current international clinical practice guidelines, for example, PCSK9, bempedoic acid, eicosapentaenoic acid, until now has not been available in Indonesia. In the future, we might also see like different methods for treating lipid, for example, vaccination and so on. I think this would be the closing slide of my talk. So the barriers in the lipid management in Indonesia actually quite multifactorial. It's not only about the confidence in terms of the physicians to give high intensity statin if needed, concerning the side effect, also maybe because they're not really updated with the newest guidelines, but also the workload of the physicians because they don't have enough time to explain to the patients that it's necessary to bring down the LDL to a certain level. It's because most cases with dyslipidemia is people did not have any complaint regarding with the LDL level, for example. And they prefer to use, for example, herbal medicines. They just stop the lipid lowering treatment because they don't feel any changes in terms of symptoms. But also in terms of available treatment, we only have statins, ezetimibe, and also fibrate. But actually, we can also intensify those treatment to reach the target, because mostly in Asian population, we need a lower dose of statin. So if we can increase the dose, for example, atorvastatin to 40 or even to 80, we might be able to reach the target. And the last issue may be about the universal health coverage, which only covers the cost of low and moderate intensity of statin, but not the high intensity statin, is also cumbersome because we cannot then give our patients those options. Okay, I think that's all, Dr. Farid. I hope we can, personally, could incite in fruitful discussion, trying to see the perspective of all the attendees. Thank you for your kind attention.
Thank you very much, Dr. Diki, for very interesting presentations and very shocking data about the last slide that you mentioned about the data from the primary healthcare of the Dr. Yuni and Dr. Wafa result. I just seen that and wow, it's interesting. For all participants, if you have any questions for Dr. Diki, you may type your questions in the Q&A chat box. For opening questions, I have actually several questions, but I would like to ask one question first for Dr. Diki about the risk model, prediction risk models that you developed for the prediction of cardiovascular disease in diabetes and prediabetes patients in the, I think, cohort Bogor. I see that the obesity is not part of the risk that include in those model because maybe it's not significant. Maybe you know the reasons why obesity is not come out in those prediction model.
Yeah, thank you, Dr. Farid. Yeah, I think it's quite surprising as well for us when we develop the model of obesity. If anything, we also see the paradox of obesity in that population actually. So even the obesity is associated with less cardiovascular event. So we need to look dig further on the explanation of that one. One of which maybe we use because we use the obesity cutoff point using the cutoff point that was developed from Japanese population, right? So might not be appropriate to our populations. But when we try to recalibrate using the data from Flores, Depok, and also Jakarta, we also see almost similar patterns. So we need to dig further. Maybe in those area, actually, maybe hypertension and also age is more dominant compared to the obesity itself. But it's very interesting. If anything, we saw the other way around. The obesity is not really increasing the risk, but I think the relative risk is a bit lower, not more than 1. I think 0.8 or something, but it's crossing the 1. So yeah, it's not significant.
Wow, interesting. Because Dr. Bambang previously showed that with increasing BMI, they found increasing of blood pressures. Yeah. And there's still no questions from the audience. So I would like to also ask more to Dr. Diki. So, and regarding the data that you've shown about the effect of the atorvastatin and rosuvastatin on the beneficial effect on the albuminuria in diabetes patients, why there's discrepancy between these two statin lowering agents? Atorvastatin have beneficial effect and the other hand rosuvastatin does not. Is there any explanation on the paper?
Yeah, it's always being asked in almost every presentation about this matter. I cannot have the detailed explanation by heart. I cannot remember by heart, but one of which is actually about relation with the fact about the chemistry of those drugs actually, but the lipophilic and hydrophilic patterns of those drugs, and also because we can see that. So in many studies actually, at 1 or 2 studies that shows that actually atorvastatin indeed is more preferable to be given in those with chronic kidney disease, because the data show that data, but the detailed mechanism I cannot remember by heart, Dr. Farid.
Okay, thank you, Dr. Diki. There is one question from the audience from the participants from Dr. Sri Suryani Wijaya. Dr. Diki, do you have any experience with inclisiran? What is your opinion about these medications?
Yeah, thank you, Dr. Farid. Inclisiran is one of the new PCSK9 inhibitor actually. but it's not really inhibit the receptor, but it's actually affecting the mRNA that lead to the production of the PCSK9. So I don't have any experience of yet because it's not being approved by the BPOM until now. I think it's just only been recently approved by the European Drug Agency for the treatment of dyslipidemia. But if you look into the algorithm, actually the role of the PCSK9 inhibitor is mostly used as a second line to reach a great reduction in terms of LDL cholesterol. Usually we use that in both people with high risk for cardiovascular disease or extreme risk and those with familial hypercholesterolemia because they usually had a higher LDL cholesterol at baseline. And based on the data that is in Indonesia, I think the higher the baseline LDL cholesterol, so the chance of reaching the target using the available drugs in Indonesia is quite low. I think from the few studies only reach about 12% reaching the target. But in that case, maybe that's because not so many of our clinicians are confident to use the high dose of, for example, atorvastatin. I also read previously, I also rarely use, for example, atorvastatin until 80 milligram. Until recently, now I'm trying to use that high dose. And it can also give a quite big amounts in terms of reduction of LDL. But if we cannot attain the reduction of LDL using the maximal tolerated dose of statin, we can combine with ezetimibe, and then if needed, not reaching the target, we can add the PCSK9 inhibitor. Or in the case with the statin intolerance, we can also try to also combine with PCSK9. The nicest about the PCSK9, about the inclisiran, because blocking the mRNA, it can also be given every 6 months, but it's quite expensive. But if it's covered by the insurance, should be good, because then it can be a potential additional drug to be used. But I think for Indonesia now, I think the first would be the use of optimal dose of statin, Dr. Farid. And in terms of high intensity statin, that should be go to choose between the Rosu and also atorvastatin, which is widely available. Thank you.
Yeah, I cannot agree more for your last statement about the increasing or the optimizing dose of those statin, because as the data shown, we are now not really use those drugs optimum or in the optimum way. So yeah, I think that's really big homework for all of us, especially in the primary healthcare. And I think also about the BPJS or the insurance, our national health insurance, how can we make them to cover this high intensity statin in the primary healthcare? That's really a big homework for us. I think one last questions or for me or comment from you about this statin use in the primary healthcare or in daily clinical practice, about the side effect itself. I think most of them are the myalgia or body ache, because I sometimes see a lot of patients experience those, especially with the higher dose of statin. Do you have experienced, are those side effect will last duration, like in the duration of they receive atorvastatin, or just in the beginning when they starting at statin, either atorvastatin or rosuvastatin?
Yeah, thank you, Dr. Farid. I think this is quite a common problem that we encounter in every day. So usually what we observe, and also from study, usually we observe this side effect mostly at the start of the treatment. So maybe it is also could be considered as well to start maybe not on the top statin dose, start maybe at a lower dose and then increase the dose of the statin. Like you mentioned, actually we saw in the ESC guidelines, we also observed that if you have like a target of below 70, we don't try to reach those target early at the beginning. First, we try to reach below 100, and then we intensify the treatment. Maybe that's one of the factors itself. And in terms for the dose, actually, if you compare there's a study from the atorvastatin 10 compared to 80 milligrams, I think the number of those experiencing the statin intolerance in terms of myopathy is almost similar comparing those with 10 and 80 milligram. I think it's quite remarkable. I don't know whether it is the same here in Asian populations, because it's not really done in Asian population, but I think it's quite remarkable to see the result of the study comparing the 10 and 80 milligrams of atorvastatin. The myopathy is quite the same. Of course, if you saw there's a myopathy, you can try to reduce the dose or stop the statin. It depends on the severity of the myopathy, and then you can also try to restart with lower dose.
Yeah, thank you so much, Dr. Diki. Since we reach to the end of our meeting today, so I would like to conclude this meeting. So our topic today regarding the cardiovascular prevention in the diabetic patients is very interesting, that already delivered by Dr. Diki, and we have still a lot of homework to do regarding especially the data from the primary healthcare about the discrepancy between the dyslipidemia and the treatment that given by our primary healthcare provider, and also the role of the national health insurance, how to optimize this into our clinical daily practice. Thank you so much, Dr. Diki, once again, and thank you also to Pfizer, Upjohn, and Novo Nordisk and Prodia for the support for this session for all the meeting today. And I would like to give the floor to Ms. Tika, and thank you for all the participants. Good afternoon. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. I think Mbak Tika has lost connections.
So maybe, oh, she's back. Yeah.
Okay, sorry for inconvenience. Thank you, Dr. Farid and Dr. Diki, for the interesting presentation and discussion. Excellencies, distinguished guest participants, ladies and gentlemen, we have had productive and inspiring time together, and this event about to come to the very end. I hope you found the presentation on this masterclass are informative and helpful. Before, we would like to inform you that we will deliver the certificate of attendance by email within 2 weeks of the event. On behalf of the committee, we apologize for the imperfections in the organization on this event. Once again, we thank to all speakers and moderators, also the participants who participated until the end of the event. See you on the next meeting. Stay safe and healthy. Good afternoon. Wassalamualaikum warahmatullahi wabarakatuh. Thank you so much.
Waalaikumsalam.
Waalaikumsalam warahmatullahi wabarakatuh.
Makasih, Mbak Tika. Makasih banyak, Prof.
Terima
Sidar.
kasih, Prof. Sidar.
Dari awal sampai akhir.